Oh, hey, welcome everybody. It's great to see you all here in person and also online. We have many attendees joining us today online. My name is Elisa Hermann. I'm head of the library here at the Museum for Naturkunde. And before we start with the actual part of the BHL Day, I will give you some more information about the agenda and the event itself. So the motto of this year's BHL Day is Bridging Data and Nature, Connecting Information Technology and Biodiversity. We will have 8 inspiring talks on innovative approaches to biodiversity data and about advancing biodiversity data through persistent identifier and data liberations. There will be 2 parts. There will be a short coffee break in between. So we have 4 presentations, then have time for Q&A, then the coffee break, then again 4 presentations, Q&A, and then we invite you for the reception at the Dinosaur Hall. We planned this as a 2-day event. Unfortunately, the workshops for tomorrow need to be cancelled. Especially to the ones organizing the workshops, we really excuse ourselves. We apologize, and we highly appreciate all the work you have already done preparing them. We'll see if we have the opportunity to, do the workshops later in the year. As you register for this event, please keep in mind that the BHL has a code of conduct. And now I would like to give over to our first speaker. Frederik Berger is welcoming you in the name of the museum. Thank you.
Thank you, Elisa. Hey. Who has ever heard of Thomas of Cantimpré? One. For those who are not familiar with the name, don't worry. Thomas of Cantimpré lived in the 13th century and probably never imagined that he would be mentioned in an opening speech of a conference some 750 years later. But before we delve into Thomas' contributions, let me extend a warm welcome to all of you. Welcome to Germany, welcome to Berlin, welcome to the Museum für Naturkunde in Berlin. It is a great pleasure and honor to host this year's annual conference of the Biodiversity Heritage Library. My name is Frederik Berger. I'm head of the Department for Service Development and Impact in the science program Collection Future. And I'm very, very pleased to have been invited by Elisa to give the welcome address for this public symposium. You're all familiar with the title, Bridging Data and Nature, Connecting Information, Technology, and Biodiversity. Symposium aims to create a bridge between, as far as I understand, between data and nature by connecting information, technology, and biodiversity. I also hope this symposium will serve as a platform for fruitful exchange and for us to address shared challenges and to strengthen our collaborations and connections. And you may wonder now how Thomas of Cantimpré fits into this context. Around the year 1250, he compiled ancient and contemporary texts and wrote a bestseller titled Bonum Universale de Proprietatibus Apud. So my Latin became a little bit rusty and I had to ask some translation devices. The abstract of this book reads as follows. In this book, in which the wonderful Republic of Bees is discussed and the general method of organizing life in good and Christian way is wonderfully explained and elaborated, a work varied and pleasant, with miracles and memorable examples from its time inserted everywhere. I think I will read the book someday, but I did not do it before this symposium. But what's interesting about it, Thomas was bridging data and nature by aggregating information sources, likely adding his own observations on nature and relating all to contemporary questions of religion and belief. His work is a testament to the power of integrating diverse knowledge sources to address the challenges of his time. I said it was a bestseller because even today, more than 100 manuscripts still exist. But most importantly, his book underwent a significant media revolution in the 15th century. It was printed only 20 years after Gutenberg printed his first Bible. So Bonum Universale de Proprietatibus Apum became one of the first books ever printed in history. And now a little bit more recently, in 2022, one of those early prints was digitized here at the Museum für Naturkunde and integrated into the Biodiversity Heritage Library. So the same text witnessed a second media transformation, this time into the digital realm. It is actually the oldest book kept at the Museum für Naturkunde and was one of the first fully digitized books during our last digitization initiative for integration into the BHL, thus enhancing its accessibility for everyone who has internet access. Coming back to the Museum für Naturkunde and BHL, the shared history dates back to 2009. The museum coordinated digitization initiatives within the EC-funded project Biodiversity Heritage Library for Europe, a consortium which consisted back then of 28 partners from 13 countries in the European Union. It's a project that nicely underlines some of the goals of the BHL, exemplifying the power of collaboration by making biodiversity literature openly available to the world as part of the global biodiversity community, providing open access to critical biodiversity knowledge. BHL addresses major research challenges. It supports the global scientific community in understanding and preserving Earth species amid climate change and extinction crisis. Since its inception in 2006, BHL has been working toward a shared future where biodiversity knowledge is globally accessible and supports universal bioliteracy. Aiming at these goals continues to be ambitious and important, and I believe perhaps it's now more important than ever before. The Museum für Naturkunde piloted the project to bring open access journals as virtual items into the BHL. This not only indexes our journals published at Pensoft, but also makes them directly accessible. And this is one of the great advantages, of course, of modern research literature. As digital-born objects, their texts are directly full text searchable, simplifying research. Additionally, they have identifiers, hopefully, from the start, integrating them into the citation network. However, as we have seen with the example of Thomas, BHL is committed to not leaving historical literature behind. They successfully assigned DOIs to articles that might otherwise have been forgotten. But even digital-born objects are not inherently reusable. Good metadata in publications are essential to extract as much biodiversity data as possible from them. And this is just mentioning some of the topics which will be addressed and discussed today. What I know for sure is that Bonum Universale de Proprietatibus Apum will witness yet another revolution. Even if the medium remains digital, the way we access the original text, the questions we ask, the knowledge we derive from it is changing with emerging technologies. And at the same time, looking back into the past can remind us of the responsibility we have as a community to preserve knowledge and make data accessible. Will people 800 years from now be able to retrieve the data we are producing today? I don't have an answer to that question. But I firmly believe that the preservation and accessibility of data is one of the most important challenges we are facing in these times of real rapid societal and technological change. Before I hand over back to Elisa, let us carry forward the spirit of collaboration and innovation that has brought us together today. Let us remember that our efforts not only shape the present, but also pave the way for future generations to access and to build upon the knowledge we create. I wish to thank David Igelden, chair of the BHL, as well as all members of the executive committee and secretariat for bringing the annual conference to Berlin. A special thanks to Elisa Hammond for the great organization here on premise, as well as to all our colleagues from administration and event management who have been involved in making today's and this week's event possible. Finally, of course, many thanks to all speakers for their contributions and to all of you online and here in the room for joining. Enjoy your symposium.
Thank you very much. And now we would like to start with our first 4 talks. The first talk will be given by Sabine von Mering and Siobhan Leachman, and they will present how the open knowledge base Wikidata can be used to link and enrich research expedition data. Sorry, it was a long day. Data sourced from Biodiversity Heritage Library. Yes.
Good afternoon. My name is Sabine von Mering. I'm a biologist and data scientist based here at the Museum für Naturkunde Berlin.
Hello, I'm Siobhan Leachman. I'm a Wikimedian from New Zealand, and I'm also a Biodiversity Heritage Library volunteer. And we're going to be presenting on how the Biodiversity Heritage Library is an absolutely vital source of data about research expeditions. So just as an overview about what we're going to be talking about, we'll be showing the importance of natural history research expeditions and why we need identifiers for these expeditions. We'll discuss why we've chosen to use Wikidata to help create these identifiers and how an international collaboration has been formed to undertake this work. We'll be discussing the importance of the Biodiversity Heritage Library and also the Biodiversity Heritage Library's data in aiding our efforts, and give information on how we use BHL data in our work. We'll then discuss the reuse of that data and then how BHL can benefit from that reuse. So why research expeditions? Well, they are a really important source of natural history specimens. However, currently, there's no global registry or identifier for research expeditions. And this makes linking the information or data about those research expeditions really difficult. It hinders the efforts to enrich the historical context surrounding those expeditions and their collections. So how do we solve this issue of identifiers for research expeditions? Well, ever since its inception in 2012, Wikidata editors have actually been collating information about research expeditions, creating items in Wikidata, and then linking those items to people, locations, publications related to those expeditions, as well as to the institutions that hold the collections or archives obtained by or about the expeditions. Now, by doing so, Wikidata editors have been creating a Wikidata identifier for each expedition added to that platform and have aided the efforts in collating and linking data about those expeditions. Now, just in case you don't know what Wikidata is, it's a sister project to Wikipedia. So while Wikipedia provides text, Wikidata stores structured data. It's a free, open knowledge base that anyone can edit. It's multilingual by design, so you can use it in multiple languages. It can be read and edited by both machines and humans, and it's easy to improve either a single item or thousands of items at one time. The data can be queried, and because Wikidata is openly licensed under a Creative Commons 0 license, the data can be reused without restriction. Now, importantly, Wikidata also acts as a hub for external to Wikidata identifiers. It can be used as a linking mechanism between other databases or knowledge graphs. And this structure, this openness, accessibility, makes Wikidata a really powerful way to create linked open data about topics and can be used to answer really complex questions based on that linked data.
A group of museum professionals working in natural history institutions and some Wikidata editors recognized the importance of research expeditions for collections and, of course, the wider natural history community. First, we formed a wiki project to help coordinate efforts to improve coverage of research expeditions in Wikidata and to create Wikidata Q identifiers or QIDs for research expeditions. we quickly realized that there was a need for a data model to ensure that data was added consistently. As a result, a working group was proposed that was later approved as an official TEDWIC Biodiversity Data Information Standards Task Group, extending the work we were doing in Wikidata and working towards an internationally agreed terminology and data model for research expeditions. This effort is still ongoing. Here you can see a list of Wikidata properties we are using to add data about research expeditions into Wikidata. Each statement added to Wikidata items using these properties should also be referenced and with links to sources of the data. And this is where BHL comes in, because the Biodiversity Heritage Library is a major source of information on these research expeditions. It contains publications generated as scientific results by and other publications about these expeditions. BHL may often contain information not just about the expedition itself, but also about the people who participated, the locations visited, the species newly described, specimens collected, and which institutions hold those specimens and the expedition archives. But a lot of this data in BHL is currently only in prose form as unstructured text. And we need to get this information, all this information from BHL into Wikidata. So it's appropriately structured and linked, also queryable and reusable.
So to give you just some examples of the type of data BHL holds, of course, there are publications, including expedition reports, scientific papers, about the actual expeditions, papers on the collections made during those expeditions. BHL can hold archival material, such as field notebooks, photograph albums, and letters relating to those expeditions. And if the metadata about these sources is placed in Wikidata, we can then link the expedition to the publication relating to it, and thus to the data lurking inside that publication. We can also add BHL Wikidata properties, such as the BHL bibliographic identifier or the BHL item or BHL part or even BHL page identifier, in order to link the expedition item directly to the work in the BHL catalogue. And that's basically providing all the information and data about that expedition. BHL also contains a lot of information on people, many of whom are expedition participants. It often holds publications written by those people, either about the expedition or the collections taken during the expedition. It also may contain biographies or obituaries about those people. Now, obviously, we want to link the research expeditions to their participants, and then in turn link the participants to the publications by or about them. And this linking helps provide a richer context about the expedition, the participants, and can also show the scientific impact of that expedition. And we create this linking by adding these people to Wikidata and then using BHL content again to support statements made about those people on their Wikidata items. And then we can link the people Wikidata items to the publication Wikidata item, as well as to the Wikidata research expedition item. So BHL again has a Wikidata property which aids in this linking. The BHL creator identifier property helps link the person to their publications in the BHL catalogue. By adding the BHL creator identifier into the Wikidata item for the expedition participant, this also helps Wikidata to act as sort of like an identifier hub. Wikidata acts as links the BHL catalogue to other institutional identifiers added onto that person's item. So it can include other identifiers such as other library catalogue identifiers, such as VIAF, the Virtual International Authority File, or even natural history institution identifiers. In fact, BHL may even have a BHL creator identifier for the expedition itself, which makes the linking of that expedition to the publications generated by it and the expedition participants even easier. Now, BHL also contains publications that provide information about the species and also the specimens of those species collected during that expedition. It may contain original descriptions of species new to science and obtained during that expedition. BHL may also have information on the type specimens collected during the expedition and used to create those original descriptions. And this is really important to taxonomy to link the species to the publications giving the original description of the species. By adding and linking this BHL content into Wikidata, we can sort of aid this taxonomic effort. This linking also gives context to the specimen gathering event, i.e. the research expedition. It gives context on how this type of scientific knowledge was actually generated. And finally, by adding references to Wikidata to the works held in BHL and ensuring those references contain appropriate BHL identifiers, we're able to ensure that those works are only just a click away directly from Wikidata straight to BHL.
And these up there are the BHL identifier Wikidata properties that help us to link BHL content on and about research expeditions to the participants, the publications, the specimen collected, and the locations visited, even the ships used, and so much more. And if the collections and the collections community and the Wikidata community continue to add such data, the outcome is an interlinked web of knowledge, a knowledge graph about research expeditions and the related entities, with BHL playing a major role in providing data that generates this web. As an example, you can see this figure that shows the Wikidata item for the Mexican-American botanist Inés Mejía, which is linked to one of her research expeditions, to the country where that expedition took place, to one of the institutions that holds collections made by her during that expedition, to the genus described using specimens collected during the expedition and which is named in honor of Inés Mejía, and to a co-author and participant in a previous scientific expedition with Mejía. Much of the data helping with the creation of these items and these links justifying this linking was obtained from publications available in BHL. So BHL really is a goldmine of research expedition data, helping to enrich information, improve data quality, but also to contextualize our knowledge about these events.
So it's fabulous that all this data can be added to Wikidata and making it easily accessible and reusable by anyone. But what can we do with all this data once it's in there, once it's in Wikidata? Well, as I've said, Wikidata can actually be queried. And we've got a query service. So we query the data and we can create visualizations, generating more insight into the research expeditions themselves. And here you can see a very basic visualization of the links in one Wikidata item for a particular research expedition. And it's telling you the places it's been, it tells you where the collections are held, who participated, and more. Natural history institutions can link to the Wikidata item number or QID in their collection management systems. And in this way, the Natural History Museum can link to the data that not only is held in their collection management system, but also to the publicly available data about the research expedition in Wikidata. And this helps enrich the institution's collection management system, but also provides context to the people actually using that system. Now, on this slide, you can actually see a screenshot from the website of Te Papa. That's the public website. Te Papa's New Zealand's National Museum. So this website shows information from Te Papa's collection management system via Te Papa's API. And as Te Papa's collection management system has links to both the English Wikipedia article and the Wikidata item about this particular expedition, these also appear on Te Papa's website. Now, the Wikidata item for that particular expedition completes the round trip of this data because it also has Te Papa's agent ID attached to that Wikidata item for the expedition, which in turn results in the Wikidata item for that expedition acting as a hub because it can connect the Te Papa agent ID to say something like the BHL creator ID for that expedition. And as a result of all this linked data, Te Papa can now decide whether they actually want to pull data about that research expedition directly from Wikidata into their catalogue, if they so choose.
Third-party tools can also use Wikidata for their own purposes. For example, the tool Bionomia empowers the linking of collectors of specimens to the actual specimen themselves. Bionomia uses the Wikidata QID for the person if that person is deceased. Bionomia then searches GBIF, the Global Biodiversity Information Facility, for specimen records to see if they can find collector names that match names used on the person's Wikidata item. An algorithm used by Bionomia then suggests matches, which Bionomia volunteers either confirm or reject. Once specimens have been attributed to a collector, Bionomia produces a profile, and this is the one showing the profile of Inés Mejía. This profile shows where and when the collector was active, the taxonomic groups and specimens collected, and gives additional information, such as co-collectors and scientific articles that have used their collections to create new scientific knowledge. Institutions can use this information provided by Bionomia to work out which specimens have been collected during an expedition, even where the specimen label fails to mention this. Another example of a third-party tool that reuses Wikidata is Expeditia. This tool uses the data and images placed in Wikidata about expeditions to visualize this expedition on a map. It searches Wikidata for research expedition items, and for each one of them checks if there's an itinerary. If there is, it can show the journey on the map and illustrate it with images imported from Wikidata. Expeditia can also show specimens collected during the research expedition and place images of those specimens on the expedition journey map. The tool does this by taking collector and specimen information from the Global Biodiversity Information Facility, and much of this data has been added to GBIF through the work of Bionomia volunteers linking the collector as represented by their Wikidata identifier to the specimens. By combining both the structured data in Wikidata and the information in GBIF, Expeditia can then provide more context and information about research expeditions and their collections.
Finally, moving on to how work in Wikidata can actually benefit BHL. Now, once the BHL creator identifier for an expedition or even a person is added to a Wikidata item for that expedition, BHL is actually able to link the Wikidata to the Wikidata identifier in its catalogue, and if it chooses, it can pull additional data from that Wikidata item into its catalogue. So, for example, BHL can add to its catalogue not just the Wikidata identifier, but also identifiers sourced from that Wikidata item from other institutions, such as VIAF, as I've mentioned previously, or other library identifiers. BHL can also import alternative names listed in Wikidata for that expedition, making the information in the BHL catalogue easier to search and find. So Wikidata can help enrich the BHL catalogue and make it, help enrich the BHL catalogue and make it easier for those who are actually using BHL to discover and explore more information on expeditions. Now, in our final slide, we would both, on behalf of our wiki and also research expedition communities, want to express our gratitude for all that BHL does. In particular, we want to thank the institutions and the people who work for those institutions for making BHL such a fabulous resource, because we couldn't do what we do without BHL. So thank you.
Thank you very much. If there are questions, we will please collect the questions, and we will come to this after the whole presentation in this part. Also, if you're attending online, please write your questions in the chat. And up next is Mike Trizna. Mike Trizna will join us online. And he will give a talk about BHL datasets as educational resources for AI training, and as case studies in crafting comprehensive AI interpretable data sheets.
Okay, so hopefully everyone can hear me there. Guten Tag. Wish I could be there right now, but the timing of this was not
Wait
great.
a second, Mike.
Okay, sure.
Okay, I think you can start sharing your screen now.
Okay. Okay, did that work?
Okay, we're good to go.
All right, great. All right, and I will go ahead and start my timer over here too. Okay. So, Guten Tag, everyone. My name is Mike Trizna. I'm from the Smithsonian Office of Digital Transformation. Even though I'm at the Smithsonian, I don't work for BHL. I work kind of alongside them. And today I'm going to be talking, giving a talk called From Machine Readable to Machine Ready, Evolving BHL's Data for the AI Era. And again, the last talk, they finished with the slide thanking BHL for being such a great resource. I'm going to kind of start off with that. I think that BHL is the perfect example data set for teaching AI for a few reasons that you're all aware of. So the fact that it's that a large majority of the content is open and has permissive copyrights means that it's really easy to kind of remix the different content in BHL for presenting to different audiences. Also, it's really great for teaching AI applications because it's a great combination of image and text. And then also the entire data set is very global and collaborative. There are so many people working on it, so it's not very weighted in one direction versus another. So that makes it great for teaching, and also it's captivating. If you've ever gone through the BHL Flickr collection, you know that the content is filled with these beautiful illustrations all over the place. They're kind of all over various different biodiversity efforts, web presences. You probably see it all over GBIF, their website, as well as BHL's website, and you find these illustrations kind of make their way around the internet because they're really beautiful. So because of that, I guess my first kind of collaboration with BHL was back in 2022. I did a presentation at the Digital Data Conference organized by iDigBio, talking about a personal project I actually did where I knew that BHL had this great resource, lots and lots of beautiful open source images on Flickr. But I was having a real hard time kind of doing natural language searches through it. So I used this as an opportunity to learn a few AI skills, and built what's called a multimodal search interface where you could actually search in text keywords and use that to find images that match those. So this is kind of a silly example here. So it's a painting of a bird in the style of Lisa Frank. If you're a child of the 80s or 90s, you maybe remember Lisa Frank binders and different school objects that were really flashy colors. And it finds birds that match that style. And that this is all on open source on GitHub, and all the code is there. That was a really fun project, and I used this to select the a few prints for my son's nursery. So in doing all that, the AI work actually wasn't all that hard, but it was a ton of work to grab all of that data from Flickr. It's all technically there in the sense that it's open, anyone can grab it, but getting all 300,000+ photos was a pretty tough effort. I will point out that now a lot of the metadata has been uploaded to Figshare, but that was after the fact. So it was a pretty tough effort. And then I also produced these vector embeddings from an AI model that was used to enable that that search there. And I, had all of these images basically sitting on my own computer afterwards. And it would be great to kind of repackage all of this for somebody else to do the same thing, and probably make the interface a little bit better and improve in the way that open source software improves. Yeah, and I was able to, the way it's split up are into albums that that represent volumes in BHL, and then from there I grab all the photos and then the metadata, and then there the Flickr machine tags that are applied by different volunteers who go through and manually apply these to the different photos. Okay, so I mentioned that BHL has a lot of open data. I tried to do like a roundup of the different data sources that are out there. It's probably not comprehensive, and I'll show you another diagram that shows the full extent of BHL data. So the major ones here are the BHL website. Itself has an API, and then hosts on the BHL website metadata exports in bulk. The Internet Archive has images and OCR from the BHL holdings. Figshare, which is I think it's going through the Smithsonian instance of Figshare, has a few packages there with metadata and OCR as well. GitHub has code that's used on supporting BHL, and then also Flickr metadata exports are there and actually connected to Figshare, which is interesting. And then a really useful new resource that's come online in the last couple months is this AWS registry of open data that hosts metadata, page images, and OCR. So like kind of all of the underpinnings of BHL is there for people to grab without BHL having to pay for any of the bandwidth or anything that AWS normally charges. And here's the full extent. This is actually, ironically, this is on Figshare itself, but this shows, I guess, all of the different transit that BHL data goes through to ingest data, to export it. I'm not even going to bother going into all of the different sources there, but you can check that out later. The takeaway point here is that it's pretty complicated. Yeah, and I guess to kind of put a point on it, the overall purpose of sharing these data sets initially was for open science and data preservation. So for the open science bit of this, I have a really old, one of the original Smithsonian Institution logos, which includes our mission statement, which is for the increase and diffusion of knowledge And this is how you know it's old, is because it used to be among men. They took that those last two words off. Yeah, and then also data preservation is, was kind of like a side effect, but I did point out Internet Archive on that roundup a few slides ago, and when that went down for a couple weeks, we kind of realized how important it is to have data backed up in various different locations. But now with the advent of AI, it's also important to have data shared out there for training data, for custom AI models, for specific tasks that are things that are being done on a routine basis with BHL data. And then also documents and other biodiversity data in general. And then it's also really useful for benchmarks, for evaluating pre-built AI models. So if you even attempt to keep up with the AI world, you'll see that like every single week there's a new, better solution for a certain AI task, like OCR or large language models or things like that. And they will put out their own benchmarks, say that it's the newest, greatest thing, but it really takes a lot of work to apply to your own unique set of data that you work with. So that's why it's really useful to have benchmark data sets, so you can say, it actually does do much better on handwritten field notebooks that are in BHL. Okay, so I'll add one more suggestion of data sets or data repositories to that big list, which is a place called Hugging Face. It's kind of got a funny name. It's named after an emoji. And the Hugging Face hub itself consists of three different major aspects. The first is the models there. So anytime somebody publishes a really important open source AI model, they will almost always host it on Hugging Face. It's meant to store AI models and also serve them, and it can be used as a way to actually kind of like plug directly into them. It has spaces, which are demo locations there, so you can actually host Python web apps there, as well as any other language, really, if you can put it into a container system. And then data sets. And this is the part that I'm going to concentrate on for the rest of the talk here. So just a quick kind of sidetrack here. So there was a really important paper in 2018 called Data Sheets for Datasets, authored by Timnit Gebru and a few other authors. She was at Google at the time when this was published, but this is a pretty major paper in the world of AI about proposing a documentation framework for machine learning data sets, because a lot of these data sets were being published and didn't have a lot of background information about how they were put together, the motivation behind putting them together, and their recommended uses. So this paper was pretty important in the field for establishing a foundation for AI governance that also led to documentation about AI models and data statements also. So Hugging Face has really leaned into these that paper. They call their data set cards, and these are a few of the different fields that are suggested as soon as you upload a data set to Hugging Face. So some of these are pretty self-explanatory, so like the license, but a lot of these are really great for giving credit to the people who put a lot of work into putting these data sets together. So there's a field for curated by the data repository. So this kind of implying that Hugging Face is not the main source for these data sets, So things can live in Figshare or live in BHL, but Hugging Face can kind of host a sample of them or an offshoot of them. The big one here is suitable and recommended use cases. So you can suggest certain things that you can do with the data set. And then you can also, in the next one, point out out of scope uses. So you can say, we don't think that you should, we recommend against using it for certain cases. So that's really, ooh, I just knocked my mouse off the desk here. And then you can also get into like the data set structure. So this is where you talk about define the different fields, the curation rationale, the annotation process. So talking about how you actually went through and annotated your data set, who the annotators are. So this is really important, especially like in the citizen science project, where you might have a lot of volunteers. You can give them credit here. And then this field is, I've found is really really intriguing when talking to people at the Smithsonian, at least. When you talk about the bias, the risks, and the limitations here, so we went through a process a couple years ago on making a few data set cards for a few data sets within the Smithsonian. One of them was on the bee specimens at the Natural History Museum, and the curators could kind of spend days telling you about all the different biases and limitations of the data set, how it doesn't represent all bees of the world. You know, it's only the ones that the collectors had access to, and they were collected mostly in the summer. Didn't mean that bees were more prevalent in the summer. It's just like when the curators had an opportunity to go out into the field. And then feedback contact, I'll point out, is really important for if people notice that something's wrong with the data set and want to suggest corrections or fixes. Okay, so by publishing a data set on Hugging Face, you also get some built-in data set tracking, kind of akin to GBIF, I would say. So the kind of the big obvious one here is you get the number of downloads, which most systems provide, but you can also track kind of like the derivations here. So this is from a sample data set where somebody went through the different AI models that were trained on that data set, and then all of the demos that are available there. You can also request a DOI for tracking there, but it doesn't create one automatically just in case you have one somewhere else, but you can kind of specify how to cite the data set. You also get this really nice data set viewer as part of this that shows kind of a distribution of the different fields, sorting, things like that. It'll actually show images in line. And then another really cool thing is that they recently released this thing called a data studio, which allows you to do SQL commands on like database commands on searching through your data set. And because it's an AI platform, you can actually kind of give it your instructions in plain text, and it will generate the SQL for you correctly, I'd say like most of the time, and run it right there in the browser. So yeah, some suggested BHL Hugging Face data sets I think would be great to have up there are the Flickr illustrations. So that was kind of what I started with some recommended use cases of being able to pull out different animals and plants from images. This is an example right here. This is kind of taken from a full page illustration with a few other things in there, and you can pull out just the bird head, OCR names and artists from the pages. You can make models that mimic a certain style of a illustrator. You can try and do taxonomic classification, and then reverse image search, which kind of folds into that demo I showed before. And then you can fully document the selection and annotation process and give credit there. And then another big one I think would be really, really cool is that the BHL Flickr dataset was generated pretty manually by BHL staff and volunteers going through and picking out the illustrations. And you can kind of see in this overview which is the one that would obviously go in there, but you can't really pull that from the Flickr dataset itself and say, you know, all of these clearly are illustrations. You actually need all of the other ones that were tagged or not tagged as illustrations to be able to train a model on recognizing full page illustrations. Also OCR and HTR, once you have all that text, you can do things like summarization, entity extraction, like names, dates, scientific names. We actually do have that as part of a BHL data set. Question answering, and then custom OCR models potentially. And then I've got like right at time here, but I've got a last slide. I wanted to point out real quick that a caveat on AI products in data sets, that you do need to have manually validated ground truth for them to be fed into custom AI models. So we have OCR in the BHL datasets. Those were generated by AI models themselves. We do have, this is a screenshot here from a blog post. We do have some manually annotated text in BHL and in other places, but this is a big project that came over recently from the Smithsonian Transcription Center of field notebooks, and we can actually use that subset as evaluation because they are validated by experts that that the text on the field notes actually matches up, and then you can actually build truly useful custom models. Here's kind of some takeaways here, and then some sources to the actual links. And I actually did use a model hosted on Hugging Face to remove the backgrounds on all these. So I'll stop it there.
Thank you very much, Mike. Unfortunately, in between, we lost the connection when you were on the slide with the dataset tracking. It was just away for a few moments. I hope we did not miss anything important, but otherwise we may come back to that in the Q&A. Our next speaker is Gitanjali Yadav, and we do have a bad internet connection today, but she luckily pre-recorded her presentation, and now we have a video of the presentation. She will talk about how natural language processing has transformative potential to enable extraction of knowledge from scientific literature into knowledge management of phytochemistry. And we have the video here. Please. Okay. Oh, and but Gitanjali will be back for the Q&A. Yes, we can.
So I thank colleagues in the museum for this opportunity to speak at this symposium on bridging data by connecting information, technology, and biodiversity. BHL 2025 is very timely, of course. Thanks for also serving as a wonderful gateway to the wealth of biodiversity knowledge around the globe, but also crucially for revolutionizing the way information is disseminated and accessed. You're playing the
video. Hello? We can't see the video at all. There's just a table. Can you replay from the beginning, please? Oh, I'm so sorry. Yes, it's working now. Please press the play button. I think it's not pressed.
Brilliant. See the slides now. Right, so I thank colleagues in the museum for this opportunity to speak at this symposium on bridging data and nature by connecting information, technology, and biodiversity. BHL 2025 is very timely, of course. Thanks for also serving as a wonderful gateway to the wealth of biodiversity knowledge around the globe, but also crucially for revolutionizing the way information is disseminated and accessed. You're playing a very vital role in advancing scholarship, innovation, and global collaboration. Open access has completely transformed our work here in India, as well as in many other places in the global south, enabling us to bypass research barriers and, just as envisaged by the BHL and the Natural History Museum. The fabulous line of talks and panels that we have today strengthens the power of collaboration, and I do hope to convey that we are the beneficiaries of your efforts to make biodiversity literature openly available to the world. My own talk today is about the phytochemical landscape, the dynamic mosaic of changing chemistry that surrounds us in space and time. Plants, particularly, have evolved some of the most advanced forms of communication on the planet. They communicate with each other as well as with the rest of the biosphere through chemicals. Unpacking these chemical arsenals is not just about pushing and merging boundaries of modern technology, but also about being able to understand and appreciate how this chemistry is pivotal to human well-being. As you might see on screen, phytochemicals are all around us, and all of life is complex chemistry in a semi-permeable bag. And our aim is to digitize all of the plant emissions that have been recorded to date using scholarly literature. With this work, we also hope to contribute to the wider Earth Metabolome Initiative and international Asia, right here. Think of the familiar bluish tinge that you see in the air as soon as you enter the Himalayan foothills, the yellow of sunflowers. Think of the flavor of tropical spices. All of these and much that we cannot see or fathom are part of variable chemical code of plants. Variability is the key word. There are more than 35,000 unique biochemicals that are produced by plants that are not required for growth or development. Isoprenoids, which are released as volatiles, constitute the most diverse group of phytochemicals known, and this is the group that we are investigating in the lab. The full complement of isoprenoids in a given species is called the terpenome, and the billions and billions of tons of volatile terpenes that are being emitted by plants annually around the globe are mediating very specialized adaptations. These terpenes have they've got a lot of industrial applications and pharmaceutical uses. They are biofuels, they are feedstock, they are fragrances, flavors, very much in use in medicinal chemistry, and so on. But coming right back to the variability in plant chemistry, it is not just in the structure of the emitted compounds that we have, but also in their function. And this is the variability that we are, which is evident at all levels, as you can see on this slide. It is in this image, you can see the variability is within and between species. It may be between individuals of the same population, between tissues of the same individual, between developmental stages while growing, and so on. So I have basically divided my talk into two sections. The first part is about how we measure or capture this variability in emission profiles using a combination of real-time experiments, but more importantly and more pertinently for this panel today, by using literature, text, and data mining. Secondly, I will move on to how we study this variability, what we do with the data that we collect, and essentially where we combine modern and traditional knowledge-based approaches. So, how do we get emission data? Conventionally, we identify small molecular weight compounds. They are usually emitted as headspace volatiles, and we perform steam distillations, GC, mass spec, and so on. For very large molecular weight compounds, we perform the HPLC, and these processes have over decades been improved and improvised in many different ways to capture the headspace in the field, not just in the laboratory, using dynamic pumps, spinnies, portable GC machines such as the Xenos, and so on. However, even under the most conducive conditions, in the best of circumstances, all variables being perfectly aligned, all the cosmic forces being perfectly aligned, there is no way for you, for me, or anybody else to experimentally analyze or measure the chemical fingerprint of each and every plant species on the planet. But what is possible is to do the analysis on a large enough scale so as to draw statistically significant patterns from the population. And our premise, therefore, is that much of the information that we seek lies already existing in the published literature. So 15 years ago, we began extracting and compiling plant emission data from literature to create the essential oil database. It's freely available today. It's got hundreds of thousands of records. It's got thousands of compounds and species from all over the world. The essential oil database is of great advantage because it is, it doesn't just provide the compound emission data, like what compound was emitted by what plant and in what amount, but also the instance of collection, what else was going on, the contextual information, right? At what time was this emission made? What was the source of collection? What was the experiment that was conducted? Where did this emission happen, or where was it recorded? The prevailing conditions for the plant at that time, and so on. So the main shortfall that we have is that essential oil database merely represents a tiny fraction of what we could potentially find. So this is the stage 10 years ago. We don't have enough data. We are not representing the entire essential oil. Come today, where do we stand today? Do we have actually access to millions of research articles? The answer is yes. We can potentially find much more in the full scholarly corpus of published data, but what does that require? That requires access. And so our first strategy is the challenge of extracting knowledge from papers. We've started by creating a gold standard, a series of codes that allow us to extract emission data from published articles directly without reading the paper. Okay, and this information is usually available as tables inside paper. So it's not in the text of the paper so much. I mean, the contextual data is in the text, but the compound emission data or the composition profiles are in tabular format. More often than not, these tables come in diverse formats. For example, here is a table for lantana and ornamental, but also an invasive around much of the world. The most critical data is the name of the compound and the name of the emitted plant, and we also need contextual information, which can be found in some of the columns of this table, but also in the rest of the paper. Similarly, here's another table with compound emission data, but for various different species, not just one. Unfortunately, such contextual information is extremely difficult to capture automatically without manual intervention, right? And so one has to revert to article metadata every time. So that's one of the challenges we are facing in automation of this entire work. The other issue is with collecting large volumes of chemical data. There are complexities associated with data disaggregation, with normalization, and careful attention for species names, for location, for geography, for plant parts, and every variable that we are collecting, every contextual data point that we are collecting, and of course, verification of that. So we are convinced that we will be able to greatly increase our repository, the essential oil database, with a search of the full scholarly corpus, where the ray of light for us came through with the help of American technologist Carl Malamud, who gave us access to a gigantic store of article metadata extracted from 73 million research articles dating back from 1847. But now we also have the open annex, right? So in order to achieve, in order to avoid breaching copyright, we've built code that can crawl over the metadata, scanning through the world's scientific corpus and pull out insights without anyone actually reading the text. And this is very doable. So in order to build code that would scrape literature or extract or retrieve information about plants and their chemicals, what we need is a set of dictionaries. And these dictionaries can help us to scan, different kinds of data sets and map terms or named entities to the papers whose metadata is being read by the algorithm. This is being done within Semantic Climate, where we are, where is our community? Yeah. So Semantic Climate is a virtual citizen science community creating open modular knowledge. We are making toolkits, softwares, dictionaries to read open published scientific literature and make it semantic, annotated, readable, and understandable by every single citizen. Essentially, what we are doing is shown on the previous slide, which I skipped. I'll take you back there. We crawl the metadata for any plant species and their parts. We have a full list of known plants that we term as a dictionary. B, any volatile compounds. Again, we have lists of dictionaries of different thousands of compounds. And C, where were they located? What was the location? Was there any specific time? Was there anything else, such as developmental state, some kind of stress, some kind of disease? What have you? The other issue with collecting large volumes of chemical data involves complexities that are associated with data disambiguation and normalization. I told you already. So what we, so all of that is also happening while we do this, and this is happening through this community of youngsters, volunteers. We are all together learning by doing, and this QR code on the screen is to let you know more about what we do, and you're more than welcome to join us if you would like, especially calling out to early career researchers who may be listening to me today. So what do we hope to do with all of this data that we have collected? In this section, I'm going to tell you a little bit of what we do. We do a lot of biodiversity informatics, of course, but today I will talk about how invasive species are a focus in the lab. One of the major side effects of the seamless movement of plants and animals that, across the world that began in the Victorian era and continued further with globalization was the introduction of invasive species or plant immigrants with no natural predators to control them on new soil. One man's flower became another man's weed, and some of the most aggressive invasives can be introduced into a very small area and then escape or explode out, competing for nutrients, lights, pollinators, dispersers, crowding out other pre-existing species, or we call them native congeners. So this can lead to species decline, species extinctions, and various aspects adding on to climate change. Some of the most disruptive invasive that have become only too familiar to many of us around the world are the Japanese knotweed or the rhododendron conticum that you see on this slide. Pretty, but severely invasive in much of Britain, whereas invasives like Lantana and Ageratum, Prosopis juliflora, which we call as the Vilayati kikar in India, are very well known to the Indian subcontinent. So what we have done is to collate as much information as we could find from the essential oil database on invasive species, and then we try and see what we can find out of that. So this work is an ongoing effort of curating thousands of chemical compounds extracted from literature, particularly for invasive species, focusing on Lantana camera and Ageratum conizoides. Several groups over the past 5 decades around the world have identified composition profiles of Lantana under various conditions and contexts. What we did was to collate all of these foliar emissions from around the world and compare them to existing composition profiles of the same species from native or near native habitats, which are again available in the published literature, right? So the people who are doing this are the heroes of our team of semantic climate. Some of my students who get this work done. Abhishek collects ecological and headspace data, really processes this in the lab. Jamila was a very short-term intern who added biotic and abiotic information for each location and converted the data into feature vectors. Ewan Salter's work, you can see here, he constructed a neural network-based classifier to demonstrate the feasibility of using plant emission profiles as unique species identifiers. And he assessed the congruence of plant chemotaxonomies and phylogeny. What he found was further evidence for the existence of multiple chemical variants in the invaded habitats of Lantana camara, one of the world's most aggressive invaders. Further providing indirect but strong evidence for the novel weapons hypothesis of invasive species evolution and success. One of the biggest advantages of having geographical emission data is that it can be mapped onto other historical or public global data sets, such as the World Trade data, and you can combine the World Trade data with the occurrence records that are there in the GBIF. So the Global Biodiversity Information Facility is a treasure trove of identification records associated with all plant species. I mean, lots of species, but definitely plants. For example, so what we did was to use the first historical GBIF record of observation of an invasive as a proxy for the first appearance of that species, of that invasive in that given location. And then we checked for existence of connecting evidence to other native or invaded regions among the records available in the World Trade data. And then we mapped the GBIF and the World Trade data onto Google Maps and around the same temporal windows to get a handle on when the species arrived at a new location. We did this correlation, as you can see on the slides now, with essential oil database records of Ageratum gonizoides and reconstructed the invasion history for this major invader across various continents. We also found further support for phytochemical expansion by looking at the chemistry, the chemical classes of Ageratum gonizoides between native and invaded habitats across major continents. Okay, so that again, as I said, provides evidence, adds to some more evidence on the novel weapons hypothesis. So these plants are indeed, invasive species are indeed using chemical arsenals. Finally, last but not the least, I would like to say a few words on the huge global breast plant potential that herbarium specimens offer to us. Meticulously collected breast plant samples that are available across herbaria all over the world preserve a tangible record of botanical diversity and have long served as a foundation for botanical, taxonomic, and systematic studies. We strongly believe, I initially worked with the Cambridge University Botanical Garden that you can see on the slide here, my favorite botanical garden because I've spent many years working there. We strongly believe that we can find triggers in the genomes of invasive species by exploring the small nucleotide polymorphisms that may have been retained in the herbarium samples of Lantana or invasives around the globe as the invader traveled from one geographical region to another. In the end, I would only like to reiterate that I hope you've enjoyed and made some sense of my talk. We are very happy to collaborate. You know, the dazzling variation in plant chemistry is a primary mediator of trophic interactions, herbivore predation, parasitism, disease, you name it. And at the same time, these interactions are also feeding back to influence spatial and temporal variation in chemistry of plants. As we investigate the shifting spatial and temporal mosaic of plant chemistry within and between species, we hope to identify genomic patterns that serve, as a nexus of these trophic interactions and nutrient dynamics. None of this work would be possible without access, but access does not just mean reading material, and I hope my talk has been able to convey that. Availability is not the same as accessibility, and therefore we all need to join hands once again and collaborate to be able to verify the data sets that we are using, particularly for biodiversity. Here is my group in India, most of my students, postdocs, interns, and we've all just completed in this picture a semantic hackathon for sharing the semantic toolkit with the rest of the community. I thank my collaborators. across the world, students and our funding agencies. And you can find out more about Semantic. Our mascot Amy is on screen right now. You can find out more about Peter's work, my work, and our Semantic climate on screen here. You can also use the QR I used earlier. This is Peter Murray Rast. Yes, and that's the first day I arrived in Cambridge. That's Wenky R there. And on the right at the bottom is a hack that we did in India together a couple of years, just before the pandemic. All righty, so with that, I would like to say thank you very much. I will stop sharing slides and check my timer. Everything looks good. Thank you. Goodbye. Happy to take questions later.
Yeah. Hey, thank you very much. Our next speaker is with us in the room today. Roderic.
So, colleagues in the museum for this opportunity.
Oh, okay. Thank you. So Rod Page will demonstrate how we can use new tools such as large language models on BHL content.
Okay, hi, I'm Rod Page. I'm one of those really annoying speakers who keeps changing the title and the topic to some extent. So this is going to be slightly different to what was advertised. The title is Future Interfaces for BHL, which is a bit of a silly title, really. It just gives me a chance to use one of my favorite quotes. The future is already here, it's just not evenly distributed, by William Gibson. So what I'm going to do today is talk about one possible future way of interacting with BHL. Now, to start with this, I'm going to try and give you some context. This is my kind of view of BHL. By the way, I'm sort of one of these kind of side people. I'm not part of BHL, but I sort of interact with them and annoy them enough to sort of be semi-part of it. So for me, there are kind of three bits to BHL. There's the community, the infrastructure, and the interface. So community is basically people, many of whom are in this room, who are part of BHL, contribute to it, do the scanning of the literature, the users, the volunteers, all the people kind of connected with that. The infrastructure is the servers, also all the sort of basic kind of software that runs it. And finally, there's the interface, which for most people is the website, for some of us is the API. Now, I think each of these has some problems. The first one, for the community, one of the sort of striking things for me is the lack of a kind of single place you can go to talk about BHL. If you compare us, say, with iNaturalist, with GBIF, two really large biodiversity platforms, or something like Crossref, which we interact with a lot as part of the publishing community, they have a forum where you can go and ask questions, criticize, complain, but make positive suggestions, share experiences, all sorts of things. There's nowhere for BHL people to do that. And I think that's a major kind of problem. There are solutions. Those three platforms I've mentioned use a software tool called Discourse. And I think it'd be great if we had a place where we could go and talk about these sort of things, especially given current circumstances. Infrastructure. We've heard a bit about this already. So basically, there are two big chunks of infrastructure I want to touch on. One, of course, is Internet Archive, where all our images and data and text are stored. Last year, that was hacked. There was a politically motivated attack on Internet Archive. It went down. That crippled BHL. One response to this is to move to other servers like Amazon Web Service. That's nice. Doesn't necessarily solve the problem, because the guy who runs Amazon makes rockets that look like dicks, and he's a billionaire. What could possibly go wrong? rocket-making billionaires do not have a great track record in maintaining stable infrastructure. So maybe we need to think a little bit beyond that. And also, the web has changed a lot. So the current BHL design is lovely. It's been around a long time. I guess probably the biggest change is the move to mobile, which BHL just really does not work. If you've got a phone, every Sunday morning, I look at BHL first thing to see what's come out that's new. It's on an iPhone. It's really hard to see. And in a sense, this is kind of ironic because the first time I ever saw an iPad was actually Chris Freeland was holding it in his hand. Chris Freeland used to work for BHL. And this was kind of like the future of the web. And it hasn't quite sort of got to BHL yet. So what I'm going to do is I am going to focus on the last of these and sort of talk a bit about having a new sort of way to look at the web interface to BHL. This is a sneak peek. I'll show you a bit more in a moment. Now, I want to emphasize this is not a replacement for the existing BHL site, which is huge, serves all sorts of people. This is a tiny little snapshot, just the kind of front end to looking at some content. All of BHL is not in here, just a small bit. So it's kind of a toy, but it's a toy that we can sort of play with. There's a QR code there. There's a link there if you want to go and have a look at it. Right, quick detour. This is one of those slides that if you're a tech person, yay, lots of interesting stuff here. If you're not, it's kind of, oh God, it's full of icons and buzzwords and all that kind of stuff. If you're a tech person, there are a few things I'm just going to very quickly point out. And I was going to say, if you are a tech person, you're probably going, maybe wouldn't have done that. PHP, really? Really? So basically, I've taken some data from BHL. I've put it in a kind of database called CouchDB. The only thing to know about that is this is a database that originated in the States, but most of the developers are now actually here in Berlin. The images, I wanted to sort of be as independent as possible from big rockets and Internet Archive. So these are on some servers, again, here in Germany. The way I'm displaying the articles uses a technique that's not IIIF. Those of you in the museum community serving images, you may be very much aware of IIIF. I don't like it, so I'm not using it. And lastly, it runs on some sort of web platform. Okay, enough of the icons and the jargon. So I want to show you some sort of screenshots of what this could look like. So the idea again here is just to show you this is a different way of looking at BHL. First, this is just a couple of journals. The one on the left is a French journal. Pretty colors. One thing I've discovered is there's a lot of content in BHL that's kind of slightly hidden because of the way it's kind of shown. On the right, this is a German journal. The reason I showed this is technology. One of my pet peeves is when we look at BHL, we focus on the pretty, the old watercolor pictures of organisms. It gives the impression that BHL is gorgeous, but old. This is a paper from a few years ago. It's got molecular phylogenies, it's got ordinations, it's got specimens. This is very modern kind of literature. So you have the complete range from the old pretty to the new sort of high-tech kind of science. There is some pretty, of course. This bizarrely is in the Birds of Australia, which I don't quite understand. The diagram you're seeing on the left, this is a journal, and what you're seeing here is when you click this list of pages, we've got each one of these is an individual article. And this is something that Nicole is going to talk a bit about in the next session. One of the challenges in BHL is finding articles within these kind of volumes. Now, there is pretty in BHL. I have a different definition of pretty. This, for me, is one of the sort of most neglected pieces of work in BHL. This is a work on ostracods, or crustaceans, that has these kind of stereo images. And there's volumes and volumes of this. And the neat thing is, if you basically do this, and then if you've got your face really close to that picture and kind of look like with a 1,000-yard stare, those two images coalesce, and you suddenly get this 3D picture of an ostracod shell. And there are volumes and volumes and volumes. Even though they've been scanned and everything, you can still get, this works much better on a phone, sort of plastered in front of your face. So we have 3D scans just kind of sitting in there. Now, there's a couple of things I've been really keen to do. One of them is to make this kind of content work on a mobile device. So this is just a little kind of demonstration of that. The screen gets smaller. Some of the bits sort of go away. There are the details. I stole this shamelessly from the Hattie Trust web interface. Some of you may know Hattie. It's basically where Google's books content has ended up there. So I kind of stole it from that. This is looking at an individual journal. Again, very kind of pretty. Works both on a desktop size screen and on a mobile kind of screen. Okay, so enough of the pretty. What can we actually do with this? So one of the things I've tried to do is to make the interface a bit more kind of useful. As much as BHL is about pictures, it's also about text. In this case, you can now select text. So the text is sitting there. You can click and drag and copy it. Here I'm using Google Chrome. This is an article in French. I don't read French. I've clicked the button. Google has translated it for me in situ on that web page. So this is the kind of experience you might expect if you're looking, say, at a PDF in a regular PDF browser or on the web. I think we should try and do that with the BHL content. Now, what's nice about having text that's selectable is more than just this, we can start to annotate it. How many people have heard of hypothesis with a .s after it? There's a few hands that are going up. This is a really cool project. Essentially, you can, with a browser extension, select any kind of text content on the internet and annotate it. Now, in this case, what I'm doing is I'm selecting a bit of text in the same article, and there's this little kind of sidebar pops in. You can write comments, you can highlight it, and anybody can do this. So if you want community collaborative annotation for free with virtually no code, this is the way to do it. You may have encountered this. Pensoft journals sometimes use this. There are a few journals which do this. So by essentially slightly tweaking our interface, BHL becomes something everybody can sort of pile in on and make sort of comments. So that's the possibility of manual annotation. There's also, of course, annotation with computers. In this case, I've written a little bit of code that goes through the text and does probably the most trivial thing you can do, pick out, say, latitude and longitude. So in this case, these are this little bit here, latitudes and longitudes. That means we can then start to put this content directly onto a map. So this is probably about 3 or 4 different volumes of content in BHL. Each one of those little red dots you can see is a locality mentioned specifically by latitude and longitude in BHL. This is a tiny fraction of what's there. I run another site called BioStore, which has many more kind of data points like this. So now we can browse BHL interactively on a map. Now, this is very early. I haven't figured out, to me, one of the big challenges with maps is how actually do you use it. The moment we can see here is you can kind of click on it. You get a hexagon that says, okay, this is where you clicked, and we can maybe get a list of all the papers in that area. I'm not sure that's the best way to do it. I think there's a lot of kind of thought to be done on what's the best way to geographically search a database like this. Okay, so you might have been expecting AI, and AI is obviously a big kind of theme. So yes, there is going to be some AI. This looks actually scarily like a Tesla robot. It's not. So one of the challenges is BHL is basically pictures of paper. And we want more than that. We want kind of get the structure of a document. And there are lots of kind of tools that people have proposed to do that. There's one that I found that I think is really, really interesting. And this is something sort of opened my eyes to what we can kind of do. There's a website called Datalab, datalab. I think that should be .io, but it's now 2. I have to check that. Essentially, you feed in images of a page, and this comes back and says, okay, this is a figure, this is a caption, this is the page header, this is the page footer. It does that in a couple of minutes for like a 300 or 400 page PDF. You can run it on your computer for free. You can run it in the cloud. And what this does is it allows you to interpret the structure of a paper. So I got a bit carried away with this. So one of the most simple things you can do is basically feed it a BHL volume and ask it to extract all the images. These are all individual images extracted from this particular volume. These are not the pages with a picture. This is just the figure nicely kind of cropped. So you could imagine, for example, the kind of things that Mike was talking about earlier about doing image search, reverse image lookup, and all that kind of stuff. Here are all these images that you could use. I did get a bit carried away. If you like shells, there are shells. Butterflies, lots and lots of butterflies. Again, it's picking out all sorts of things. Pictures of butterflies, obviously not a butterfly. Graphs, charts, all kinds of things all sort of nicely kind of come out. Weevils, like weevils, this is a monograph that's about 100 years old. Absolutely gorgeous illustrations of these insects. And all this is automated. This is just a few minutes processing for each volume. You've got all the figures. Now, I could show you many, many more of these. The reason I'm so excited about this is because it gets us sort of closer to part of our goal of being able to sort of get information out of BHL. So what you're going to see here, there's another project, and Donat's going to talk to you a lot about this later on, called PLASI. Now, many of you are probably familiar with PLASI. PLASI essentially works by taking essentially mostly the modern taxonomic literature, born digital content, born digital PDFs, extracting information, extracting figures, information on species descriptions. Now, in some ways, PLASI's task is, I won't say it's easy, but it's easier because a modern PDF like this one on the left often comes with built-in information. It tells you this is a figure, and so you can write code to extract a figure, and away you go. We don't have that with BHL. We just have a picture of a piece of paper. But with this tool, we can now start to classify elements on the page. So what you're seeing on the right, this is the same page from the same article, and the sort of faint colors there tell you that down the bottom, we have, this is a figure caption, this is the actual figure, this is some text, these are some headings, this is a heading for the whole page. That's all done automatically. So we are, in many ways, very close to what PLASI does on the left, the similar kind of page structure that they've extracted. So this means that we could do very similar things to what PLASI do for the literature that BHL has. The next step would be to say, okay, that's the text, what is it saying, what are the latitude and longitude, what are the specimens it's mentioning, and so on. So I think this brings us much closer to being able to sort of get meaningful data out of this content. Okay, so where next? What's going to happen with this? So the first thing I need to say is it's a toy. It's a proof of concept. I'm just basically saying this is one way to think about reimagining how we could display BHL. And it's a small, tiny bit of BHL. I've tried to select a bunch of things. The example you can see there, that's a field book. I've got some monographs, some old stuff with the pretty, some more recent sort of taxonomic papers and so on, just to sort of experiment with how this works. One way to think about this is a test bed. It's very low risk. So it's like you have this very well regarded, highly used website at the moment. You probably don't want to mess with that too much because it works. But one way to sort of explore new ideas is to say, okay, we'll have a separate little thing. We can play with it. If it breaks, it's not a problem. But also then you have freedom. You can start to think, okay, is there any other way to display this content other than the way that sort of seems sort of obvious? And perhaps the final kind of thing is maybe another way to think about this is you could have sort of mini BHLs. In the same way, for example, GBIF has recently released portals, little kind of individually sort of set up snapshots or views into bits of GBIF. And again, we're going to hear a bit about that this afternoon. BHL could do the same. You could imagine, okay, let's just have a BHL or a view of BHL for a particular country or a particular species, or maybe for a particular person, expeditions or something like that. And of course, we've been here before. There was a time where BHL had BHL China and BHL Africa and BHL Europe, and I'm sure there are probably others. Sorry, Nicole, there's no BHL Australia on this. And these have all basically gone away and failed, which is why BHL Australia isn't there. So you could imagine these are all separate platforms or separate sort of projects with their own kind of code. Another way is to have the same code with just different content. And maybe we could sort of bring back this idea of multiple kind of, the BHL that you particularly kind of want. So that's all I was going to say. There's a QR code and a link. Have a look. It's live. Be gentle. And as I say, it's just a fluid kind of little sort of toy to sort of explore, what could a different kind of way of exploring BHL look like. Thanks very much for your time.
Thank you very much to all speakers. We are way ahead of our schedule, which gives us plenty of time for Q&A. I just check, are there questions already in the chat, or should we start with the room? Okay.
Sorry, quick question, if it's possible to share the slides.
Yes, we can share the slides on BHL Day website after the event. And in the chat, some folks are sharing their slides already for users online.
Thank you so much.
I do have a few in the chat for earlier speakers in the day. This one is for, I do believe, Siobhan and Sabine from Nikki Nicholson. Do you intend your project to also cover contemporary fieldwork expeditions? If so, have you considered privacy implications regarding individual participants and where they conduct their fieldwork? Is Wikidata set up with appropriate procedures to handle this?
Okay, this is Sabine speaking. We have decided, or we have discussed, that the project shouldn't just focus on the historical expeditions, even though a focus of our current work is on those historical ones. We are aware that some groups, for example, in Norway, are already using Wikidata identifiers to compile and collect metadata for recent field work. I forgot the name of this series. Do you remember? No. I have to look it up. So it's series of vessels going into the North Sea somewhere, and they are using Wikidata for this. But as far as I understand, with a limited restricted set of metadata collected and information collected. Obviously, putting information on like recent field trips, would be critical and difficult, and we're not doing this at this point because you could track some like reoccurring itineraries or places where people are going. So we're not doing this at this point. However, we believe that it would be useful to have identifiers to collect at an earlier stage before the knowledge is lost about information like which people went, where are their permits, but possibly some of this information should be inside of institutional repositories for a certain time, but we have no solution for this at this point.
Thank you. Other questions?
I have another online for Siobhan and Sabine. From our other panelist, Gita, thank you for a wonderful talk. My question is about your very impressive expedition knowledge graphs. Can these graphs be downloaded in any specific formats, XML or SIF or CSV? Thanks a lot.
So yes, you can query Wikidata, and yes, you can download it in those types of formats. There's a particular tool that we use called Scholia. It is a Wikidata tool, and you can then basically search for research expeditions and then download the results of the queries in those different formats, or you can create your own Sparkle query about the information you're wanting to extract from Wikidata and extract it in those formats if you want.
There was one question from Rod.
This is a question for Mike. I don't know if Mike can hear us. Can you?
I can.
Yeah.
Can you hear me?
I can hear you fine, Mike. One of the things in the AI world is this idea of kind of like a leaderboard of scoring. And I'm wondering, GBIF, for example, has challenges every year. Could you imagine a sort of BHL AI challenge where we sort of score different models for, say, the ability to recognize images or something like that?
Yes, absolutely. I think that would be something great to organize around. I forgot to put in a plug for another organization I'm a part of called AI for LAM, AI for Libraries, Archives, and Museums. And that group is putting together different working groups based on different tasks, and they're doing exactly that for audio transcription, which doesn't really apply to BHL. That's probably why I didn't bring it up here. But they, you know, there are really good open source AI models that will take audio recordings of oral history interviews and transcribe them, but they don't necessarily do so well on old scratchy recordings that are in museums. They're benchmarked against people with their iPhones or YouTube videos or things like that. So they're putting together this data set that's a little bit more representative of what people in museums or libraries have. So I think BHL could organize something similar for, you know, for field books or for other types of documents that, you know, a lot of people are trying to tackle with AI that doesn't always do as well as it's advertised, so that you could actually be very standardized in saying, you know, it doesn't do so well, and then measure it against each next release of an AI model.
Any more questions online? Yes.
I have another online. Hello all, I'm curious to know if illustrations are or can be directly tied to research expeditions in addition to specimen images and Expeditia or anywhere else. Do you think these illustrations be useful as visual proxies for specimens that do not photograph well 100 years later, such as fish and marine invertebrates?
You're getting definite nods from me. Yes, 100%. Yep. There is ways to link in Wiki. We've been uploading a lot of BHL images directly from BHL and also from Flickr into Wikimedia Commons. Then you make a category, you link them via the category to the item, the Wikidata item relating to that category. You add structured data, which is an ongoing process at the moment. It's relatively new, and there are ways to link it all via structured data to ensure that you can actually link the image to the research expedition. Wikidata doesn't necessarily have items for specific specimens because Wikidata is big enough and there's too many specimens for that. But certainly in relation to research expeditions, that's definitely possible.
That was the question from the chat. Okay, do we have any more questions? Yes, don't.
I have a question to Rod. So this is where these tools are very intriguing to use hypothesis or a data lab. And they're really interesting and promising. The question is, so the expensive stuff is actually not to get quick image sorted out, but it takes somebody to look at that and say, okay, that's correct. And that's a lot of effort at the end. So how do you envision to preserve that? And how do you envision to actually add this annotation to the BHL so it's preserved for eternity?
Good questions. I mean, I think you can do this in stages. I mean, you could easily have, these are a whole bunch of images extracted. Nobody's looked at them. You take them as they are. But also we can then, so the next step for me would be, there are tools we can use to then check, is that bit of text a caption or so on. So I think we can probably get an enormous amount of mileage through automation if we accept some error rate. In terms of the annotations and hypothesis, live in hypothesis. We could back them up if they want. They'll be there as long as that survives, or we can make backups of that. And again, because anybody can annotate, you could then have a discussion within the annotations if somebody disagrees with a particular annotation. Likewise, it would be kind of useful. So I showed the latitude and longitudes extracted by a computer. If the computer gets it wrong, you could, an hypothesis, put an annotation to the effect, this is wrong. So I guess it boils down to what level of error rate is going to be acceptable for what you can get out, given the resources that we have.
No, so the question is not so much the error rate. You could probably reduce it quite a bit by having 2 or 3 people looking at the same. But assume these annotations are helpful and promising, and you wanted to use them for something else. How do you preserve them?
Well, we could basically, there's an API, we could download them and store them someplace. That's just a kind of data storage kind of question. I mean, there are various annotation platforms that are being sort of experimented. We saw one yesterday that BHL was playing with. The reason I particularly like Hypothesis is it's large, it's widely used, it's been going for about 10 years. So that gives us some degree of hope that it's going to be around. But we could easily download every annotation made in this way and stick it somewhere like Zenodo or somewhere it's going to persist.
I've actually got a question for Mike. One of the things I find challenging is I'm very interested in scientific illustrations, and particularly the illustrators who do the scientific illustrations. And I noticed in your presentation, you gave an example of show me all the images in the style of this particular illustrator. Is there any way to do something similar instead of in the style of this illustrator, to actually say, I want to try and find illustrations by this particular illustrator, not just via OCR, but also, I guess, the style of the illustration itself or the publication it comes from, whether you know the illustrator was illustrating that publication and automatically.
Oh, I was ready to answer it, and then you threw in that last part. That makes it a lot more difficult because so much of the, I guess, those machine tags in or not machine tags, I guess they're called machine tags, but they're kind of applied by people in Flickr, were kind of like done following some kind of detective work. But yeah, I think there's definitely some room for doing what you're talking about of kind of like matching styles, but maybe also of I'm not sure if Rod was like getting to this, but I think there's a lot of uses for AI to make suggestions that can be validated by people. So, you know, types of patterns that are like, you know, this volume, we were able to automatically pull the illustrator name from the plate in like 80% of them. If 20% are unknown and they look the same, you can kind of make the assumption that it is the same illustrator. So again, to go, I hate to do this, but to go back to the audio transcription project I mentioned before, we're finding that it doesn't get 100% accurate, but you're able to get like 95% accuracy rates in audio transcriptions, which are a huge benefit to hand off to somebody over what was traditionally done, where people actually had to listen word by word and type out every single word of the transcription. So a lot of these kind of things that give something to people to correct rather than start from scratch is a big benefit.
Yeah,
So I'm not sure if that's what you had in mind.
No, that's exactly what I wanted to know, because I was one of those people who spent hours and hours and hours doing those tags. So looking at pretty pictures and doing tags, and if we could have found a way to do it a lot quicker, a lot more efficiently, and you just give me 5% that needs correcting and automation takes care of, or, you know, the computers take care of the 95%, I would have been a very happy woman. Yeah, so thank you.
Yeah. But, and sorry, this is a little bit more than you asked for, but to get to something that Rod asked, I think there is this tricky bit there of like trying to track like what actually came from a person and what came from an AI suggestion. You know, you can have an AI suggestion, but like, should you preserve it if the human annotator overrode it and said, no, I found that this actually came from a different illustrator, or like in a handwriting transcription that it really is this word instead. So it's I haven't seen like a great perfect solution for like keeping track of all of those like suggestions and overrides, I guess, for annotation. So I think that's something that the AI world is kind of working on together.
Lisa, I'm going to take one back here.
Hi, this is JJ for a question for Rod. IIIF is becoming a widely accepted image sharing standard on the web, and I was wondering why you don't like it, and also if you have any other open source viewers with strong community support and development support for sharing images online. Obviously, we are looking at IIIF, it's widely adopted by GLAM. So I just want to make sure, like, if we do develop a BHL pilot, that we're using the right tech to do so.
Oh boy. Okay. The short answer is IIIF sucks, but the longer answer is there's two bits to it. There's the standard for describing how you present information, and that's okay. It's a bit over-engineered, I think, and it's getting better. I've gone through various versions. It's getting there. I guess my issue is more, partly the viewers are awful, but secondly, I think it's the perspective is wrong. What they do is they kind of center the image. It's all about an image. I can zoom in. Most of the tech is about serving big kind of tiled images so you can zoom in and zoom out. And I don't think that's what we actually want. But I think, despite the pretty pictures, most information is in the text. And I think that what you're doing is by using IIIF as it's currently implemented in the viewers, you're treating BHL as a set of pictures of paper. And if we still think in terms of pictures of paper, we're not going to get to, you know, the holy grail of extracting information from text and stuff. So so there are two bits, and my gripe is really with the implementation of the viewers rather than the underlying metadata. I can live with that. But I just think there's better ways to kind of do it.
Okay. Are there any other questions? Otherwise, we could already go into our coffee break, and we'll be here for 3:45. Correct, right? Okay. Have a good break.
Hello and welcome back. I'll be your moderator for this afternoon session. Thank you all so much for staying and giving us all your attention and time. I am Kelly Try. I am the Biosciences Librarian at the University of Illinois, Urbana-Champaign, and I am Vice Chair on the Biodiversity Heritage Library Executive Committee. And I'm really excited to be introducing our speakers for this afternoon. So first up, we have Nicole Kearney, who's going to talk about persistent identifiers and other accessible tools for finding our information and data.
Sorry, I didn't know. Why doesn't it work? Oh. Oh. Okay. Yeah. Presentation.
Hello, everyone. It's really lovely to be here. I'd like to thank Elisa and everyone from the Secretariat and the BHL for organising this wonderful event. I'm Nicole Kearney and I manage the Australian branch of the Biodiversity Heritage Library. I also lead BHL's Global Persistent Identifier Working Group, and it's that work that I'm going to be talking about today. We work with a number of different persistent identifiers, but today I'm going to focus on DOIs because we've now minted over 50,000 of them for journal articles on BHL. Before I begin, I would like to acknowledge the First Peoples of Australia because that's where I live and work. These are all illustrations that accompany the very first published descriptions of Australian species. This is a time in our history where the animals and plants of Australia were being discovered by Europeans. And the text that accompanies these illustrations very rarely contains any acknowledgement that there were already a people in Australia who had an extensive knowledge of both the biology and behaviour of these species. So why are DOIs so important? A DOI, or a digital object identifier, is a unique and permanent identifier that provides a persistent link to a piece of online content. I started obsessing about DOIs about 10 years ago when I realised that while over 90% of newly published articles in the sciences had DOIs, very few of our historic articles did. And this was a problem. DOIs for modern literature are assigned at the point of publication, and they can instantly then start being used wherever those articles are cited or mentioned, such as in this 2018 tweet announcing the discovery of a new species of pygmy seahorse. That DOI provides a persistent link directly to the primary literature. And if I click on that, I get taken through. And if you're interested in pygmy seahorses, you can scroll down to the reference list where you'll find more DOIs. And each of these DOIs links you directly to the other publications online, providing this great linked network of scholarly research, and you can link from article to article in a never-ending chain of knowledge. But there are articles on this list that are missing DOIs. And so the readers are much less likely to click on those because who can be bothered finding an article if there isn't a link to it these days? Which is a shame for that article because it's on BHL. So without DOIs, articles are much less likely to be read and readers and authors are much less likely to cite them. So the journal articles on BHL were in danger of falling into obscurity without DOIs. The other thing that BHL was missing without DOIs for its journal articles was metrics. So this cute little seahorse has a very impressive outmetric wheel that tells us exactly how its DOI is being cited and shared. I wanted to cover BHL in these rainbows to prove that the historic literature is still extremely relevant today. So in 2020, we started a persistent identifier working group to retrospectively assign DOIs to the journal articles on BHL. Team RetroPIDs, as I like to call my group, brings together the incredible knowledge and expertise of BHL's librarians, our programmers, metadata specialists, persistent identifier experts, and BHL super users. We've met fortnightly for the past 5 years, which means that the members of Team RetroPIDs are as persistent as our persistent identifiers. So thank you to all of them for everything I'm going to share with you today. So our overarching goal was to bring the historic literature on BHL, the foundation of all our biodiversity knowledge, into that modern linked network of scholarly research. And to achieve that, we needed to do 2 key things. The first was we needed to assign new DOIs to the articles in BHL that didn't have them, to make these articles discoverable, citable, and trackable. And we also needed to add external DOIs to BHL articles, particularly external DOIs that resolve to paywalls elsewhere. I'm going to talk about both those things. So what's involved in assigning a DOI to a journal article? Well, first you need somewhere to point the DOI to, and that was our first problem. BHL is primarily a library project. We digitize bound volumes and we upload them online along with their volume level metadata. So this volume contains the very first description of the platypus, and the article data, sorry, the data that was attached to that volume was just this volume level metadata. So the title of the series, the Naturalist Miscellany, the author of that series, George Shaw, the volume number, and the specific date of that volume, 1799. And that's it. No information about what was contained within that journal volume. So in order to assign a DOI to this journal article, we needed to first gather and upload the article data. When you upload article data into BHL, it automatically creates a beautiful article landing page, complete with all that article data, a link through to the content on BHL, and a downloadable PDF. The other thing that happens when you add article data to BHL is that it generates a table of contents within the book viewer, which allows readers to navigate from article to article. It also adds the title of each article underneath the title of the series, and you can click on that to get through to that article landing page. So most importantly, we now had a specific URL for each article to point our DOIs to. And so the final step was to mint a DOI. And thanks to Mike Lichtenberg, our lead programmer, that's very easy for us to do now in the back end of BHL. We just add the articles to a list of articles that need DOIs, and within 24 hours, they have one. And those article DOIs are then added to these article landing pages. So this was one of the very first articles that we assigned a DOI to as part of this project back in 2020. And this is now its Outmetric wheel. So it's definitely a rainbow. And every DOI on BHL has one of these Outmetric wheels, which means that we can track the ongoing reach and impact of this research. So if I click on that Outmetric wheel, I get through to a report, which tells me exactly how this platypus DOI has been mentioned and shared. I'm going to run through some of the highlights of that report. Firstly, it's been mentioned in 257 Twitter posts, now X, from 169 users. That's reached over 6,081 people. Sorry, 681,000 people. It's now starting to appear on Blue Sky. I'm not really using Twitter anymore, so I'm excited to see that it's appearing on Blue Sky. Over 17 Twitter Blue Sky posts from 8 Blue Sky users. Again, that's reached 26,000 users. That DOI has also been mentioned in 4 blog posts and on 10 Wikipedia pages. Having a DOI really helps with Wikipedia because Wikipedia has this really handy automatic add a citation function. So all I had to do was drop our DOI into that box. Wikipedia automatically generates a beautifully formatted citation, complete with our DOI, which obviously provides a direct link through to the content on BHL. So I'm not really a very clever Wikipedian, but all I had to do was drop that DOI in the box and to create a citation for the illustrator of that beautiful illustration. So having DOIs on our articles helps Wikipedians make our content discoverable. Interestingly, those 10 different Wikipedia pages that now cite that DOI appear in several different languages. So we have Korean, Vietnamese, Indonesian, and French. So making it easy to add BHL citations to Wikipedia really broadens our reach. Outmetric also provides a list of the publications that have cited this DOI, which tells us that it was cited most recently in July last year. This is that article. It's all about fecal bacterial communities in the platypus, which are apparently really important for its conservation and management. When I scrolled back a bit further and look at some of the older citations that had referenced it, I found that the DOI had been cited in an article published in 1999 and another in 1995. And this was over 20 years before we minted the DOI for this article. And I thought, how is it possible that the DOI is appearing in these old citations? It turns out that the publishers of these articles, Oxford Academic and Elsevier, regularly rerun their citation indexing. And so what that means is that our retrospectively assigned DOIs were being retrospectively linked to old citations. Now, this was super exciting for me. And so I went down a rabbit hole trying to find more examples. So let's start with the most viewed book on BHL. So this is the 1758 edition of Systema Naturae. We added a DOI to this title back in 2011. So we've been assigning DOIs to titles on BHL for quite a long time. We've now assigned over 146,000 DOIs to book titles and journal titles on BHL. The Outmetric report for this publication tells me that this DOI has been cited over 3,000 times in academic papers. The most recent of these are from the future. So this DOI is going to be cited in June, July, and August of this year. Those are either preprints or they're in press, which is super exciting. And if you go back, this particular citation is cited about monthly, going back in time, which is pretty nice. Scrolling back way into the past, the oldest citation picked up using this publication's DOI via this citation tracking is from 1940. And here's another example. BHL assigned a DOI to Audubon's Birds of America in 2012. The earliest citation of this publication picked up by our DOI tracking was in 1858. So an 1858 article now has a DOI telling us that it cited our publication, Birds of America. This is that 1958 article that cites Birds of America. It's on SpringerLink, which is a commercial website. SpringerLink assigned a DOI to this article, to their copy, which makes their copy the definitive version of this article, and that's the version that everybody else must cite. Now, this article is well and truly in the public domain, but it's pretty much a free-for-all when it comes to assigning DOIs for out-of-copyright literature. Basically, SpringerLink got in first. That makes their copy the definitive version, the one everyone must cite, and they've put it behind a paywall. So if you want to use this article or read this article, it's going to cost you €40, which is a little annoying because there's an open access version on BHL. So this brings me to the second really important role of my persistent identifier working group, adding external DOIs to BHL articles, particularly those articles that resolve to paywalls. We've now added over 70,000 external DOIs to journal articles on BHL. Now, a DOI is a critical part of a bibliographic or a bibliographic metadata, which means that we should include that DOI wherever those articles are cited or shared. So it's actually best practice that we include them and add them to BHL. But when we do this, something really clever happens. Algorithms searching for that DOI can now find our open access versions. And that means that when you go to this paywalled article on SpringerLink, because we've added that DOI to our metadata, there's this little unlock symbol that appears in the corner. And that tells us that Unpaywall has been able to find our open access version. And that's again because we've added that external DOI to BHL. So I'm assuming that most people in the audience will have heard of Unpaywall, but in case you haven't, it's a massive open database of 52 million scholarly articles. And there's an extension that you can download which will provide those instant links from the paywalled versions through to the legal open access versions. This is another significant piece of biodiversity literature behind a paywall. It's the very first published description of the leadbeater's possum from 1867. Again, clicking on that green unlock symbol will take you to our open access version on BHL because we've added that external DOI. Now, the leadbeater's possum is particularly close to my heart because it's the faunal, the animal emblem of my home state of Victoria in Australia. It's also critically endangered, which means that open access to information about its past distribution and abundance is critical to conservation efforts. And these external DOIs that we're adding to BHL are essential for providing free access to this important information. Before I finish, I want to share some of my personal DOI highlights, which include assigning DOIs to the species descriptions in the very first published book on the flora of Australia, a specimen of New Holland published in 1793. We've also added DOIs to all the descriptions in the Zoology of New Holland, which is the earliest publication ever published in the world dedicated to Australian fauna. It's also the very first publication to use the word Australia, so it's very significant for that reason as well. And we've also assigned DOIs to every description in every volume by John Gould, from all of those publications from Australia, as well as those elsewhere. Now, Rod mentioned that we're all focused on the old and the pretty. That's pretty much me. I love the old and the pretty, but we do not play favourites when it comes to DOI assignment. So here are some DOIs we've assigned to articles about cyanobacteria and marine fungi, which you might argue are actually quite beautiful in their own right. We also just don't assign DOIs to old publications. So in Australia, I work very hard to get copyright permission for in-copyright publications. We digitise material in Australia on behalf of 62 organisations across the country. Many of them give us their most recent published articles. So here is an article with a DOI on BHL, our BHL DOI, so we assigned this for June 2024. So this is from Australia's Journal of Botany, which is published by our Royal Botanic Gardens. Here's another DOI that we've assigned to an article published in November last year. This article is from a journal of plant systematics. So again, quite a big prestigious journal in Australia. And this is our most recent BHL DOI, which is assigned to an article published just last month in the Castlemaine Naturalist. And we're just about to receive the April edition, and we will assign a DOI to those as soon as we receive them. So what I've shared with you today represents an enormous amount of work, and I'd again like to thank Team RetroPIDs for making all of this possible. I also want to thank two of my BHL Australia volunteers, Bob Griffith and Heidi Griffith, for their ongoing 5 years worth of helping us add article data to BHL. It's actually partly because of these two volunteers that this project exists. When we first locked down in Melbourne for the global pandemic, they rang me over and over again saying, how on earth can we continue to volunteer for BHL from home? I said, I don't even know what I'm doing at home. And we couldn't access our library materials. We couldn't access our scanning equipment. So we switched to making the existing content on BHL more discoverable and bringing it all into that linked network of scholarly research. And here we are 5 years on. We've defined over 401,000 articles, and we've assigned DOIs to 52,000 of those, and we've added over 70,000 external DOIs, making all that open access material discoverable. I want to finish with some quotes from Donald Hoban, who's the previous director of the GBIF Secretariat. So GBIF's been mentioned quite a number of times today, the Global Biodiversity Information Facility, and he's also the inaugural director of the Atlas of Living Australia, which funds the BHL Australia project. So he's a fairly important person in my life. He said just last month, I see BHL as one of the top 3 biodiversity resources in the world alongside GBIF and iNaturalist. He emphasised that BHL is the access point for most of the literature that gives context to all other biodiversity knowledge. The DOIs that we're adding to BHL make that knowledge more accessible, more discoverable, more usable, more shareable, and obviously trackable. Thank you very much for listening.
Thank you very much. All right, next up, we will have Donat Agosti discussing some more information about unlocking data and the Disentis Roadmap.
So thank you for giving me a chance to talk to you here. I also take my liberty to talk a little bit differently from what the title says. At least I start differently. Who was following the CBD in '92? Okay, one. Who followed the OECD Mega Science Project? Which became GBIF in 1999. Who was at the inaugural reception of the EOL launch in Washington 17 years ago? Maybe one Rob can also put his hand up. Who was at the inaugural technical meeting of the EOL in Woods Hole, also 16 years ago? Rod and me. So what have them all together? So what's common between all of that? So in 1992, it was the first event where global biodiversity the crisis has been recognized on a global level as a problem. So it was the biggest conference at that time where number of head states were attending. Even Bush, the American president, went down there to Brazil to sign or to negotiate and to sign, but they didn't ratify it at the end, the CBD. So why do I mention that? CBD is about change. We have a biodiversity crisis. That means we have to measure, we as a scientist have to put the voice to the change of biodiversity. There's Article 6 and 7 talks about monitoring and talks about biodiversity. In 92, we would not know how many species there are, and we focused on birds, feathery things, woolly things, and some plants. In 1999, when GBIF started, the idea was that in a couple of years, we create a catalog of life. So we have this global reference system, everybody recognized that we know all the species we know already. So we have a reference system. We then can actually go in the field, tick off, like the bird people say, species disappear, there's some new species. When we started the Encyclopedia of Life, the belief was that we could do this, contribute to that, what has not already been done. So we scan our libraries, make our library digitally accessible. Today, do we have it? No. We sit here, we discuss, how can we make this information accessible? How can we scan? Why don't we get any money? In a sense, we're our own enemy. We don't care about these things making data accessible. We scan books, we scan publications, we assign DOIs, we create citation networks, but we don't care making this happen. We are sort of in a very stupid situation now that we have fantastic tools like all these LLMs developing, these huge companies sucking in all our information. We could make all our information accessible and give our biodiversity information a voice. But we don't, because we continue to publish PDFs, or we don't care about making this really accessible. And what we need really to do is we need to assemble this information. So we have to get these publications together in form so we can, then we have to make sure we keep them. So we need data sovereignty. We have to keep it, not somebody else. So we need make sure all the things we scan, we have, is in our hands. The new publication with closed access, we have to grab them, make them accessible. We have to annotate them. We have to actually make sense out of this stuff. We have to say, here is a species, and that's a block of text which is clearly defined by the scientist who says, here's a treatment, here is a name, and this we need to make citable. We have to take it out and make it so that everybody can go back and check. If you talk about a species, a holotype, and the holotype is linked to a specimen in the collection. So we have to be able to go make this link from a text who somebody says there is a holotype. We have actually to go back and check this out. So we want to go from holotype in the collection through the material citation to the treatment, to the publication, actually check. Did the author say that? This is building trust. And if you want that our information, our knowledge is trustful, we have to be able to provide that. Otherwise, it does not work. So Platz is in this business. So Platz is said from the very beginning, 17 years ago, to BHL, the important thing is that we actually link to data in publications. And we started to develop a small NGO in Switzerland who started to make this happen. We started to collaborate. We wanted to make sure that anything we do has long life. We started to collaborate with Zenodo because at that time, 10 years ago, 12 years ago now when they started, they said, we help the little scientists who cannot manage their own data by depositing their data. We thought, hey, this is great. So we can put all our PDFs in Zenodo. They said, PDFs are not data. And a lot of these PDFs are closed access, so it's also complicated. But we found out we do something different. We go there and say, we are not interested in the PDF, but we are interested to know what's inside. So we started to talk to them and create a data site type treatment. So now each streamer can be given a DOI. Each figure that is taken out can be given a DOI. And they can be linked to the PDF, closed or not. But in metadata, you can add an element that says cited by. And the treatment can make a link, this is cited to that. And you can actually add more. You can go look at, and that's sort of the different thing from just going quickly and use hypotheses and others to make annotations. The point is, annotations are expensive, they're important, they're only relevant when you can rely on them. You want to go back to them, you want to cite them. So you want to be able to go from GBIF to this hypothesis thing, material citation, and find it, look at this. We also want to be able to annotate this and say, this is a material citation, and this refers to this specimen which has been digitized. Okay, so this is my thing about what Platzi is. So, but I should talk about the Disentis Roadmap. And the Disentis Roadmap is a little bit a child of Platzi as well, because 2014, we had one of these not so successful, some people thought, kind of weird groups that talked about biodiversity knowledge graphs. Nobody would understand at that time. But one of the outcomes was that we created this Bucher Declaration on open biodiversity knowledge management. It was a meeting where we had a fantastic celebration, but a commitment by over 300 individuals and institutions to say, we will make our data openly accessible. And it had an impact because if you got the EU, then it also went also along the line, all the data they generate is to be open. So last summer, we had like a 10 years anniversary of that. I wanted to look back and saying, so what happened? And some of it is very nice. So DiSSCo is completely open, sequences are open, but literature is not. Most of our taxonomic literature is kind of closed access. We don't really have access to that, and there's very little EU and other grant support to actually open up our publication. And you here have also the problem that funding is drying out to expand this important thing to actually liberate data from publications. So we set in Disentis together and created this Disentis Roadmap. Currently, it's signed by about 100 institutions and individuals, and you can also sign it. So Disentis Roadmap was a consensus, and the idea was, to actually come up with this vision by 2035, that's in 10 years. The power of biodiversity knowledge from research publication will be fully leveraged within an open science framework. So again, open, but the power of publications, so data in publications. So this is the vision. And the specific goals are that all the major data funders will enforce that really it's open access also to data in publication and to publications. Publications shall be machine actionable. So they shall not be PDF, but they should be in a form that can be reused. Ideally, they're annotated. So you know the core elements in it, and machines can use them. And so already we have about 45 journals now that operate this way. So we don't need to discover or do something new, but we have this workflow. And you see later, that's actually very nice development that we can get this data in the morning and the evening, it's all this disseminated. So all the kind is AI ready stuff. And then we need also allocation of money for research infrastructure. So making data accessible is more than just having a free PDF. It's essentially building infrastructures that support the annotations, that support the treatments and figures that we extract and make citable. So it needs investment into this. So this Disentis Roadmap is based on the belief that the power of publications is in the data in publications. So we have to make it accessible, citable. And so that means you have to take a PDF or any article, can be Linnaeus or not, then extract the metadata, of course, and the references, we need images, tables, and treatments. And tables are increasingly important because in our literature, a lot of the data we have is in tables. So you've seen Gita's table with all the chemicals. It's always a name and a lot of chemicals. In GBank accessions, it's a taxonomic name, often a collection or whatever specimen ID, and the sequence is used. So if you actually are able to get these tables and extract that, then we can make these data points also accessible in GBIF. Plus, we can go from GBIF back to the table, back to the article to have the context of that. So when you take this out, I mean, it just doesn't mean we create a dump. We have to actually create what today is called fair data. So we have to make the data findable. accessible, interoperable, and reusable. So together with Zenodo, we created this data type, which includes all these elements, the metadata, the licenses, and everything. So there's about 700,000 treatments there. We have like another 300,000 in the pipeline, which need to be curated. But that's one of the very big things. So now we can cite the data in publications, and we can do the same we do the same for figures and for tables. And this is the very new, since last couple of days ago, the new interface to the biodiversity literature repository, a community in Zenodo. So it shows you there are like something close to 200,000 publications, more than 600 treatments, 740 images, and 20,000 tables, which are low, but they just currently are being uploaded. But the interesting point is the number of views and the number of downloads of that stuff. So clearly, this data is used. So it's not just a funny thing. And the important point is, again, so we can go to GBIF and look at the holotype deposit from a natural history museum. We find out, oh, there's a material citation deposit, there's no occurrence, and they can go back, actually, step, you can find, okay, can I trust this? What is the source? Is it right? And then you can go back to the article. Plus, the publisher also get the feedback, because then the publisher says, oh, somebody discovered that here, and I get another hit. And we can do that because each of these annotations has a unique identifier. So you create an annotation, and there's a UID, so we can actually go back. Like in this case, we can go from the GBIF record, we can go back to the phrase here. You can see the bold part, which is highlighted in the treatment bank, so you know exactly what went into GBIF. And you can also immediately go back, because these are small data sets, these articles. There is a system in place that you can say, this is wrong. You click this button, and we get an email, and this can be corrected, or you can learn how to correct it yourself. So the point here is we have to go beyond the citation network of publications. I think that's fine, it's interesting, and it's cool, but the value is to create a citation network of facts, of data. And if you look at one single material citation, the richness of links that can be made. So it's very rich. So if we can actually make this happen, and we can also create bidirectional links, not saying this is the specimen, link it to MFN, but from MFN link back to this material citation, then we really start building a knowledge graph. We can actually add all this together. And they can also do other things. For example, here you see the collections in this community. So you can make, for example, a Linnaeus collection. That means everything which is about Linnaeus is in BLR is accessible. And it's accessible because the name is hyperlinked and is annotated, and you can find them. Similarly, you can do other things like, or you can actually make a profile of a journal, say, show me all what's on this journal in BHL and BLR. Another value is that once we have the data annotated, we can make cool things. So most recently, we had the hosted portals in GBIF. They're about, I think, 30 now. So it's EJT as we first, and BDJ came, and others. So you essentially can show your journals in the context of the GBIF data. Laurence will talk more about this. And again, and then you can also look at interesting stuff. You can say, look, this is who studied this, who collected this, who identified it. So you can actually start to make really interesting stuff which you have, and also always you can go back if you don't believe in. So the other part which is convincing for the DiSSCo Roadmap is that once you have the data, we can get it to reuse. I mentioned before, we are trying to build this catalog of life. And we don't want to wait years that if somebody publishes something, it shows up in catalog of life. And thanks to the more recent trend from DiSSCo and collaborations, we now can assure that the moment you publish something, within less than 24 hours, it shows up in the catalog of life and it's in GBIF backbone. Within a week, it's part of a GBIF backbone because they refresh now, I think probably at a weekly rate, their catalog of life, or the extended catalog of life. Another point is that in the blue one, in Biodiversity PMC, we can take these publications and add them to PMC, the PubMed Central. I guess everybody knows PubMed Central, that's the repository of life science literature. But it does not include ecology because there's a decision not to do it. But now we have a way to do it. So we started to upload first treatments. And now there are about 1 million deposits, 1 million articles of treatments in this Biodiversity PMC that can be searched using the same tools as use of PMC. So it opens up our literature to the big life science community. On the other hand, the life science community has access to our data. So if you download all the PDFs you have in BHL, we can make this all accessible. So you would have immediately 400,000 more points to data sets or publications to search. And then, of course, you can build tools on top of that. So if you want to go that way, we have to think really, what is the minimal thing we need? So we need vocabularies, we need the services to find and cite this material, we need repositories to actually store. So we need to talk to GBIF or maybe to do Zenodo, maybe create our own repositories. So eventually we need to come up with a political will that says, hey, this is important. This is a global infrastructure. We need that. So in the framework of the Disentis Roadmap, we envision to build a libroscope. A libroscope is like a microscope where you actually look into a detail and get data out, or a telescope and you get it in the form, or a satellite, and you get data out you want in a form that you can reuse. So if you actually build this, we can go find the publications, we get them, we convert them, we annotate them, we make them fair, and make them accessible. So that would be a cool tool because you could say, I have this expedition, give me all about this expedition. We have the library, we go and scan the stuff, we have the tools to annotate, we can store all the annotations, we can make all the stuff, the data accessible. So what we need to be also strategic about this. So we need to kind of build corpora of target cited publications. So we need to come up with groups of scientists say, this is, I'm interested in, I want to do this expedition. Or like IPBES says, we want to get all this information on global biodiversity change and get it together. So you find them using OpenAlex and tools, you get all the publications, you put them in a form that you can start mine them. And we have to come up with what we understand is the minimal, like digital accessible knowledge. What is what you really need that our data becomes part of this big world. So this is in a way we could envision how it goes. So we have like here from analog, we go from analog to digital accessible knowledge. And these are all the steps. And we can more or less define what are the products we need to go from one step to another one. And these specifications are important because going back in the OSA process is extremely time consuming because it percolates all downstream and the go back is time consuming. Similarly, scanning can be done in different ways. So we have to come up with minimal standards to scan. So we can do OCR, or if you want image recognition, do that. So these are all the steps needed to do it. So the lessons learned over the last years, and also from the Disentis discussion, is we have to change the way we publish. We can't afford to publish tomorrow any PDFs anymore. We should stop, just don't. And if you can't, find out, make it at least structured so it's easier for machines to extract it. We have to build trust in our data by publishing open, fair data. As I mentioned, every record in GBIF should lead us to exactly this part, in the context in the source publication. We have to become a part of projects. We have to be part of the global assessments by IPBES. We have to be part of research projects like using traits for predicting climate change. We have to collaborate. We do different things. You're very good in scanning, making things accessible. Others do different things. So we really need to work more together because we don't have many resources. For sure, we don't have the billions spent in AI now. So we have to be able to see how can you actually collaborate? How can you go forward? We should build as much as possible on existing infrastructures, at least at that moment. Probably at some point we need to find funding for our own, but we have to start. And more important, and also very importantly, we have to communicate what we do so the scientists know, and we have to train the next generation. So we have to run training courses and around the globe to make really people aware and teach them how they actually can use not just the conversion, but actually use the data, which is stuck in these publications. With that, I would like to thank everybody for attention, not falling asleep after the lunch. And okay, thank you.
Thank you. Next up to talk a little bit more about FAIR data and the European Journal of Taxonomy on the GBIF portal will be Laurence Bénichou.
Thank you. So I'm going to present you the new hosted portal on JB for the European Journal of Taxonomy, and particularly how it helps the journal to switch from openness to fairness and to go beyond openness. So we operate in a context, a specific context that you all know. We've made huge effort collectively to digitize and make accessible lots of millions of pages and millions of taxonomic treatments, yet not all of them are available online and even fewer in open access. The challenge for a publisher now is to go beyond open access and try to reach fairness. The difference between openness and fairness stands in the I, the interoperable thing, which is not so easy to do. But what is at stake really? From the moment the specimen is collected in the nature, and then it's brought back to the museum or botanical garden to be studied or to be preserved, then the researcher is going to analyze, compare that to what is already known, and then describe either a new species or make taxonomic revision, and it's going to be published, which make it valid. The time it takes from the moment it's collected in the field to the moment it's published is on average 21 years. 21 years, it's the average time that it takes from the moment it's collected to the moment it's published, so it's valid. So the challenge for us is trying to reduce that time, and the only way we can do that is try to reduce the time that we analyze the past literature, thanks to more accurate and massive access to the data. And from a prospective point of view, as Donat said, we shouldn't publish in PDF anymore. We should now make sure that our data content within the paper are accessible in all relevant databases as soon as published. So this is the challenge we have to do, and make sure that basically every information in the article is pushed, all the data content in the article is pushed in all the databases that is interesting. And the best way to do that is to push that into all the partners, Treatment Bank, GBIF, Zenodo for preservation, make sure that all of the items in the article has a persistent identifier as well. So how do we do that? By semantically structuring the article. So let's take an example with a paper recently published in AGT. So this is the first page. It's obviously all the metadata. You know what metadata means. So automatically we are going to tag that metadata and make sure that we can extract the article title, give all the identifier that we need. This is very common stuff for all of us. But we try to go beyond simply metadata and try to go into semantic taxonomic structure. So here we are going to isolate all the information content in the paper and make sure that they can be pointed or linked to the information in the databases. So here we are going to say this is a taxonomic treatment, meaning all the information about that taxon in that description, including the type material, the specimen cited, the figures, the caption, could be the key as well if there's a key in the taxonomic treatment, all the bibliographic reference, all the taxon name. So everything that can be linked should be linked in that page. So how do we proceed and how do we do that? Very quickly. This is a production workflow of AGT. The only thing here that you that should be important in the context is the fact that it's the journal itself that structure and tag the content. We do that, of course, using tools, so it's automatically done, most of it, but it's control, so the quality is there. Then we send all the article, the paper to Platzi through the API, and then we get back all the annotation, the taxonomic annotation, the specific annotation for the domain from Platzi. And then we publish the paper and send it back to treatment bank for fairization. So once the publication process is done in XML or in PDF, but in XML, it's better for perspective point of view, but Platzi knows how to do it from a PDF as well, but it's not as accurate as it could be in XML. But yet then the paper, the publication is putting into fairization data, thank you. And then it helps you to be able to reuse that in all the databases that has been cited. So it could be PMC, could be GBIF, could be Wikidata, whatsoever. So now I'm going back to the AGT GBIF Hosted Portal. So once we do all that and we push and we annotate all the information in the journal, we wanted it to be visible for the journal itself. So because we've done all that job, but we couldn't say to our author, to our stakeholder, here is the job we're doing. So now, thanks to GBIF, we can be able to show that. So two important things to understand here in the GBIF Hosted Portal. So the occurrence means an occurrence is all the material cited in an article, while the data set in that particular context is an article, meaning all the material cited in the article. So there's two important features here. The first one is the dashboard. So here it's the visualization tool to showcase all the statistics from the journal. So it could be statistic from the specimen, could be statistic about the holding institution, or could be also about the type status or about the category of the status or whatsoever. We do have another important feature, it's the data. So here you can have, for instance, different views. So this is the map view. So here you have all the results, so all the material cited in the journal that has coordinates. So here you can have all the coordinates of specimen cited into the journal. You can, of course, filter that. So here I filtered that with the institution, all the specimen with coordinates that has been deposited into this specific collection. Another view is by table. So the table here is all the scientific names, all the occurrences into the table. You can choose whatever filter you want as well. You can organize that with all the row you want, so it's customizable as you wish. Then you can have a link to the specific record that is going to detail the record by itself. You have an overview here. It's the related view. So the related view, it's all the clusters. So here it's the link with all the occurrence from the journal that went to, which is linked to another occurrence in GBIF. So for instance, you may have one point here that go back to a record that has been provided by the museum. So you can have a bidirectional link from the journal to another occurrence provided by another partner in GBIF. Here is the view of the data set. So it's all the paper by paper. And if you take just one paper, for instance, you will have all the statistics of this specific paper, meaning all the metrics on that specific paper could be all the occurrence cited on that paper, or the data richness of that paper, or the number of occurrences, or the number of material cited of whatsoever. You can have lots of statistics, and you can, of course, download anything you want. So it's really very important for us because it shows not only the statistic in the journal and be able to make visible all the richness of the journal, but also it can be linked to from the journal and to the journal back then. So the benefits are multiples, very various. One of the first one is the fact that all the data are controlled by the journal, of course, because anything that goes wrong to the process can be corrected by the providers. It's, of course, an enriched information that is made available to all. But I would say that the most important thing for us is that it's going to increase the entry points to the article, and it can have a multiplier effect on citations, which is kind of what a journal look for. So it also enables journals and the community to find new indicators and to find new metrics more relevant for our field. We still have some little things, so some little issue to address. So 3 of them. Linking treatment means that all treatment citations are annotated. So we know that we do have lots of literature and in BHL, for instance, but all of them do not went to a treatment because the taxonomic treatment itself is not annotated. Linking specimen means that all the specimen is digitalized and provided with a persistent identifier, which is not the case yet, but hopefully DiSSCo will provide a unique channel for accessing that. And the last ongoing process, and particularly with BHL community, is the linking all the references. So thank you very much, Nicole, for all the work you're doing with your team. It's still an issue for us to link that. So cross-ref DOI at the article level should solve that in part. It's more easier for us to go and fetch the information from cross-ref than from the APA of BHL itself. So it's much more easier for the journal. And I think that's it. So if you want to go to the particular hosted portal, I would encourage you to go and give us some feedback. Tell us if we forgot any data that wouldn't be there, or if you would like to engage in a conversation with us. Thank you very much.
Thank you. Finally, we'll have Patricia Mergen talk about another GBIF portal, the DiSSCo Flanders portal.
So good afternoon, everyone. So I will indeed also show an application of the GBIF Hosted Portal, more in the context of collections that are coming more and more online with all their associated data, and also in the context of a local application to meet those user needs, more specifically about the Flemish region of Belgium. I think it's next on this. So for those who might not know yet about DiSSCo, a bit of context. So it's a research infrastructure at European level called Distributed System of Scientific Collections. It is about as well life science, but also earth science collections that are in the museums and botanical gardens, but also university collections in Europe. For the time being, we have about 300 organizations who committed to participate from 23 countries. It will become an European research infrastructure, which is quite a long process at EU level, where you start with an idea, then you get on a roadmap, where you have to prepare transition each time with EU funding and from the member states. And we are now in 25 arrived almost at the end of the DiSSCo transition project that is preparing all the official documentation to become an official European infrastructure about collections. The first stage is submitted with 8 EU countries who have committed to finance it in future, and a 9th, which is Italy, who promised to join soon. And we are still lobbying for more of these EU countries to join financially to become operational 26 or 27. So it's much centered about the so-called digital specimens that wants to also follow the recommendations to be a fair digital object, where you have the specimens with all sorts of linked information about the morphology, biochemical, genetic information, but also a lot of other metadata, and of course also the taxonomic data, so that it can really be a unique reference material to document our planet's history in terms of natural sciences. Here it's more the geeky representation from the colleagues when we had the iStick project, which was first implementation projects, which shows in the center the digital specimens with all the DOIs, with the JSON serialized representation and all the different links, so that it becomes really a fair digital object with all the interlinking information about that specimen. DiSSCo also wants to do e-services that they divided in core services and additional services. You can go watch under that link a very nice recent video they did about it. It composed of loan services, of collection digitization dashboard, of specimen data refineries, and help desks, annotation services, and many more services. Specifically with our group, we are looking in having a crowdsourcing system for the collections and so on. At Belgian level, for those who know the country, we have quite some different funding bodies, and we are also involved in EU projects. So we managed with quite some effort and lobbying to have all the Belgian natural history museums, the Africa Museum, the Botanical Garden Meissen, but also all the university, both on the Flemish and the French-speaking side, working together in one project, and we share our funding as best as we can. Everybody has access to all the services, and we also have the amateur natural zoological society and so on, so that we can also have users, plus all smaller botanical garden and arboreta in Belgium that are participating with their collection and knowledge. DiSSCo Flanders is the part financed by the Flemish authorities, where our goal is to enhance the access to the collections situated in Flanders and integrate them at European level with DiSSCo. We started almost at the same time as the DiSSCo roadmap I showed before in 2018 with first setup project without funding during 2 years to make a proof of concept and mobilize the collections. Then we had one 4-year project that just finished, where we set the scene by doing the inventory of the collections, where we used what was developed within the Synthesis EU projects and collection dashboards. We also looked into enhancing the accessibility in best practices on collection management. We implemented crowdsourcing platforms, machine learning, testing, and also engaging more Belgian and European participants. And just beginning of this year, we get the good news that we got an additional project funded, where we have 9 funded partners and 12 associated partners in Belgium, where we will continue with rich collections, where at the Flemish level we have almost 6 million specimens. There is a lot of expertise there. We will focus on education, collection-based research and application, and also link with the international research infrastructures. We want to enhance the management and the access and the data integration, develop and expand the DiSSCo services, address also a lot of ethical and legal challenges, which also has an impact when publications, for example, are closed source or not accessible, where we also look a bit how to make them also available in that context. And then, of course, contribute with many partners at the European level to work together with them and not work at all as a silo in a region. We really want to integrate with other services. This is just to show shortly the different work packages of our projects. And here is the impact we want to achieve on the interdisciplinary research, promoting open science and first principle, more accountability and ethical transparency. So we got really a request from our government to look more in depth in knowledge security and also to bring in citizens. Now, all I showed here, it is really important that there is a link to the literature and the citation for almost all the components I showed here for the project, and certainly the model of the digital specimens or collections, because the collections and the specimens become exponentially more available and a very fast speed. There is a lot of information connected to these digital specimens where people want trusted references of the source of the information that comes notably from the literature and other places where it's published. And as you could see, our government was injecting quite some funding in this project and agreed also The partners agreed to allocate even a lot of in-kind resources to this project. So the funding and the governments, they expect meaningful KPIs. And one of the best way to demonstrate the usage of this large information and collections becoming available is to give them the number of citations that are found back in the literature. And for that, we looked what GBIF is doing already before they had the hosted portal. We already have quite a few of the DiSSCo Belgian partners who are providers to GBIF. And we encourage also those who don't provide their data to GBIF to really do so within the DiSSCo project. And we also offer help desk together with the GBIF Belgium node to provide the data to GBIF. And what people really appreciated in GBIF is this citation module they have, where, for example, for the herbarium of the garden, we can easily see, and it's regularly updated, how many times we are cited in the literature. And if you click, you see the different publications where the data has been cited. But actually, this is linked to this DOI you see there for the paper, where they actually know that there was a reference made for this paper that the authors downloaded more than 305 million occurrences in GBIF to produce that article. But if you just go look at that article, it is not so easy to see which specimen of the garden have they used now in that paper. But with this GBIF tool, if you scroll further down, you will find your provider, and you know immediately that they have used some 379K of specimens of our herbarium in that article. It might look trivial, but this kind of information and showing the increase of such metrics is very important when we do our annual report to our government. So we cannot just say that many specimens have increased in GBIF, but we can also show that it has been used. For the moment, with that system in terms of numbers, but we would also like to have a bit more context how this collections were used, where of course it's then much more interesting to work with the collects and also some of the system you saw in the two previous presentations. And also a question that came is when we have to show to our funder what we have done, they would like a dedicated portal or website where they can see directly, okay, I don't want to see this whole GBIF thing, that's nice, but I want to see with the funding we have given you, we want a nice portal where we can directly see where are our collections. And that's why this GBIF hosted portal was really interesting for us, and that we decided to install one for the DiSSCo Flanders hub. And so from the beginning in 21, when they had pilot phases, my colleague collaborated with GBIF to make first prototypes of the GBIF hosted portal, and the whole service was launched for everyone in 22 to have really a customizable local portal where you can show in a target way what you have done in specific projects, or some GBIF nodes also use it. So in the context of DiSSCo, our colleagues from the UK, notably with NNHM, were also setting up this prototype with the GBIF hosted portal, and we in Flanders did it too. And we really try to convince at DiSSCo level for the other countries and other regions to also go down that route by showing the added value of using these systems and not really reinventing the wheel and reinstall your own system when you have something that works very well and meets your need. Here is the website of DiSSCo Flanders that you can have a look after with the link. And what we really liked is that you can have the data shown and the specimens like they are at GBIF, but you can also add your own pages by describing your partners. We have a literary module that I will show. We could also add our crowdsourcing platform. We can add the project outputs and make news, a link to our help desk, and so on. It's also multilingual, so we can apply automatic translation with DeepL also from the GBIF system. For the moment, we translated it in Dutch, but we might also add French, German, and other languages. So that's also really a quick win and added value. So here, for example, you can see the specimens of the vertebrate collection of the University of Ghent that you can directly find back with different criteria. We also installed the literature module that is quite similar to what Laurence showed in the previous talk, and where we find also very interesting that we can have already some metadata and content. So we have our own branded site. It's what's really quite easy to implement. You don't need really high IT skills, and GBIF has a good help desk. And then for the literature module, we have this automatic update. So we already saw that we have in Belgium more than 2,800 citations for the collections that are already online, and not all the partners are already in there. So it's already quite a nice number. We can have these KPIs for our funders. We can search by literature type and also how it was used at GBIF. So you can really see, has it been downloaded or has it just been cited or mentioned? So they have different levels on how it was used. So you can really see, is that just someone who clicked and looked at it, or is it really someone who used it and downloaded it and processed it further? So you can have more context, and they also have very handy topic keywords so that you can easily find back the literature more targetedly and in your local context. So as future step, we would like to have more context beyond the citation that is already on its way to do more text and data mining. We want, of course, to do further collaboration with Plazis and Nodo and set up e-publishing groups. We are also looking with the funding we have now for the next 4 years together with GBIF to maybe enhance further the services of the hosted portal, and we also collaborate with GearCycle. We very much encourage the other DiSSCo partners to use this GBIF hosted portal and to have trusted literature sources. And with the digital specimens, we would be very happy to also include what we saw about BHL in the last days here. And also Wikipedia is really a nice source, and also work more with what we have seen, what the journals like EGT and Pensoft, to make best of use of all these services and really not start again something else, and really try with the nice funding we managed to have with these different groups to work together towards a common goal. And then I still a message from Tedwick, Biodiversity Information Standards, where there is a standard called TAXPUB, Taxonomic Publication, which is quite outdated. I think I saw on the website of Tedwick that maybe 2010, 15, there was some movement, and they really would like to see if there can be a revived interest group around taxonomic publication in Tedwick, that our group could, someone could lead it or trigger it so that there are more ties between the taxonomic literature standards and all the different groups. So they would be happy if that could be done so that we can work more together on that level. One of the challenges we have is that as well GBIF as BHL, apart from paleontology, is not addressing earth sciences, while in DiSSCo it's clearly also the large earth science collection, mineral rocks, meteorites, and so on, boreholes that are really important collections. And they would like to have something like GBIF. When we have meetings, they are always jealous about what's happening in the biodiversity world and that it's not the same yet in geology. So there is the GeoCase portal, which is quite nice, but which would need more development. There are also discussions going on with GBIF that they could accommodate more geological data. Tedwick has recently released a Darwin Core extension for mineral, and the ABCD EFG, the Earth Science extension, is also out there a long time. So there is a lot going on, and it would be good to make the effort. Speaking personally from the Africa Museum, we wanted to be part of BHL, like Meise is, and one of the reasons our director refused is because we have a huge department in earth science, and he said if they don't take earth science as well, and I have to pay another one to take my earth science literature, I'm not gonna pay 2 times. So if BHL would take also earth science, he would have accepted to provide the literature. So I don't know if this is feasible, but something to discuss. So at the end, I would like to thank really my co-authors and also all the colleagues who are too numerous to mention them all, but all this could never have happened without the collaboration on a Belgium, but also international and European scale, and the different groups and associations that we really want to work together. Fine. Thank you a lot for your attention.
All right, now we'll open it up to questions for folks in the room as well as those online. We have one online already, it seems like.
I do. We have quite a few questions from our online audience. This one is for our first speaker, Nicole. Which norms regulate or mandate the use of the first DOI assignment to an article, for example, by a publisher, as the definitive DOI? How are disputes resolved if they occur? I imagine that you wouldn't necessarily be aware of existing DOIs minted by some publisher for a historical article.
The answer is that you shouldn't assign a DOI to something that already has a DOI. We are Crossref members, which means we adhere to Crossref guidelines, and they very clearly state that if a publication already has a DOI, you should never assign another one. So for those examples that I showed behind paywalls, sometimes people say to me, why don't you just mint another DOI? It's absolutely not allowed to do that. So we do not assign a second DOI. We always use the existing DOI. It does occasionally happen that a duplicate is created. Often commercial publishers, we take in a lot of external DOIs and we do find that they have duplicates. It is then their responsibility to deduplicate those DOIs and choose one as the primary and redirect the other. So it does happen, but there are processes for deduplication. But yes, it's our mandate as members not to do that.
And I have a follow-on to that from another question. To what extent does BHL keep track of DOIs that don't persist?
Well, I gave a talk yesterday for the BHL community about just that. So just this year, BHL has become a safe harbor for DOIs that have been abandoned. It's quite common with journals if they cease to publish. So we talked about mycotaxon yesterday, which is a journal that ceased publication last year. They actually came to BHL to ask if we could take over looking after their content, but also their DOIs, because they no longer could pay for Crossref membership. So those were all transferred, thanks to the work of Diane here, over to BHL, and we now manage those DOIs. We also just earlier this year took over a journal that had its website has just completely disappeared from the web. Those DOIs had all been broken for over 6 months. We've now redirected those with permission from the older publishers to BHL. So we've taken over management of now over 5,000 DOIs that were sadly abandoned, or their publishers could no longer look after them.
I'm going to ask one more DOI question from the online audience, though it may not be for Nicole, maybe others within BHL. From Nikki Nicholson, thinking about the DOI labeling of data sets as referenced in Mike Trizna's talk, will you build links with GBIF regarding their citation tracking mechanism so that use of BHL data rather than title or article scoped uses can be tracked and reported on?
Who would like to tackle this question?
I guess the problem there is BHL doesn't really have any data, I think, in the way that perhaps the question means. If BHL was to extract specimen data from its content and put it in a GBIF, then yes, that link would exist. But as it stands, there's no BHL data or very little BHL data in GBIF.
Donat, did you want to speak to this as well?
Just comment to this DOI continuous issue. So Crossref is issuing DOIs which are issued by the publisher. If you have a journal that is abandoned, an old journal, there's no publisher. Therefore, there's no one responsible who can or should or so assign DOIs. DOIs can be assigned also from data side, not Crossref, independently of Crossref. So there are a lot of institutions now start to assign DOIs to the articles because they can't afford to go into data side, in the Crossref. Crossref, I'm not completely sure where still today, but very recently does, for example, not cite articles in data site, which are not Crossref. So it's a problem actually that we don't do that. And there's another question of what is an original article? Is it, there are different versions. There are annotated articles. So a lot of the old articles in collections, they're scanned from reprints. No, not preprints, reprints. So you have all these collections of all these separate, and I don't know. They're annotated by the scientist. So should they be the same as an original, the original print, or is there something different? So there are a lot of questions around that. And for sure, there will be always multiple DOIs for the same things. So the important point is that whenever possible, we make sure we can cross-link and we find out, right, an infrastructure or so that make sure here is a DOI which is also used there.
Thank you. Oh, Nicole? Yes.
There's kind of 2 points there. The first one is the reason we use Crossref. It is the registration agency for publications. So it comes with a whole lot, to quote Rod, the chocolatey goodness that comes with Crossref DOIs means that we get that reciprocal citation linking. We get that great linked network of scholarly research. If you are a publisher and you're a member of Crossref, you have to put DOIs in your reference lists, and therefore you have to provide the links to the other articles, and therefore they get that citation tracking, which is, you know, the currency of publishers and authors. And the second point is that I've forgotten what the second point was now. I'll leave it there.
As a publisher, I just wanted to say that I agree with you. We have to go to Crossref for DOI for all the publications. So DOI, when it's related to publication, should be a Crossref DOI. When it's related to data or other things, should be on data site. It's best practice.
My second point was about duplicate DOIs for the same thing. So Crossref has really strict rules about what constitutes another different copy. So for example, if a publication in a journal is then republished in a special edition at another time, so it has different publication date and different page numbers, etc., because it's in a special edition published another, like a year later or whatever, then it can have a separate DOI. There are also cases, I mean, we try not to duplicate content that we upload onto BHL, but for example, we have digitized the whole of Charles Darwin's library, which is a special collection. And the publications in Charles Darwin's library are, we often had duplicate copies of his publications, but he had annotations if he wrote in the margins of all of his books. So those copies are kind of significant in their own right because they have annotations from Charles Darwin in them. So you might give a DOI to his copy of a book because it's his copy and it's special. In the same way that we have coloured editions of really old rare material and we have an uncoloured copy. It's the same publication, but we might give a DOI differently to each one of those. So I've spoken to Crossref at length about this. They say you've got to have a really good reason for applying a duplicate, effectively, DOI for a publication. Otherwise, no duplicates.
Thank you. Do we have more online, Colleen?
I do. I'll give a chance to the room, though, if there are others here.
Oh, yeah.
Thank you. I had a question about Plotzy, about how you're getting the treatments and the metadata about the treatments into Plotzy right now. And given the historical literature and the tens of thousands of ways that the data is presented within different journals and different articles, do you see a role for something like artificial intelligence to help pull that information out?
You mentioned an interesting point. It's the multiple ways to do things. So individual records you do manually. So you convert the articles and you can't get the metadata, do it manually. The more advanced, more modern literature, journals and so on, you create templates where you define what parts of the article are what. So you can actually automate this. So, and then still you use QC to figure out whether it's a name or not and so things like that. And we use I mean, it's very broad, what's AI, but at the end, the important thing is that you can trust what you get. And it's not so much important. And we work now for 15 years, almost 20 years on developing algorithms to do that. I'm sure there are much better ways to do it, but we just developed this algorithm, refined them by an iterative process, by finding out errors and going back to the system and find how we can refine that. And there's a lot of old stuff we just have to do, even go out beyond the article itself and find out what is the metadata.
To go online, yeah.
Sure. If you'll hand the microphone back to him, I have a question for Donat from Christina Morgan. Are we able to identify funding or grant leaders and political allies worldwide to address the political and financial support required to prioritize this data access of the life sciences? Are there working groups or advocates voicing these concerns to non-scientists?
No. No, we work on that. So that's part of this Disentis Roadmap, is really to get people interested in that together to find out and make a case that this needs to be done. So we need to talk to EU funding. I mean, and that's a long process. You've seen the timeline DiSSCo. It started somewhere in 2015 or so. And now in 2006, we are sort of creating this ERIC. It's a long shot. So we have to work on that. And we try. And we try locally in Switzerland. You're in a competition with others. You're in competition now with this Ukraine war and budget constrictions. We have another thing now with the US, which will probably also have an effect on our budgets. So yes, we work on that. Plotzy is trying to become like a national infrastructure. I've been very close, but it's We failed last time, we were in the next program, and clearly we need to work together and try. But there's not an institution who advocates literature, rather the opposite. Nobody does it.
While you have the mic, I'll give you one last one from the crowd to online. You've described the Disentis Roadmap as a consensus. How globally representative was the group, and were there people from different career stages involved? How will they be included as the work develops?
It's not globally representative. We had people from like India, the US, Europe, I think one African. We were 50 people who were involved over, some of them, quite a few of them in 2014. So no, we are not, and we don't pretend to be globally representative, but we believe that we are open for other opinions. We are open, and we understand, I think, this world of publishing quite a bit. So we are not new, and yes, we are not. And the goal, I think, is not that we are really a representative and make everything to be representative. The goal is really to have in 2035 our stuff open. And there's not one way to get there, but there are multiple ways. And I hope others copy it, do it better, be more balanced and all of that. I think, and we don't want to be the one doing it. We hope we can inspire others, because it's a huge task. And Laurence and others showed already, so we can do little things now, but you have to scale up. It's a huge task. So yeah, go work.
Other questions in the room for any of our speakers? Do we have more online? Okay, I'll go back to Colleen.
I do, thank you. This one is for Patricia from Nikki Nicholson. Is there a roadmap available for the hosted portal program? This would help people starting projects make informed decisions about whether they need to develop a web presence themselves or invest in developing components for the hosted portal program.
Well, first of all, the hosted portal program is more a GBIF initiative. So as far as I know, initially it was made for the GBIF country nodes to have a local portal to show their GBIF data in a more targeted way for their users. And then they expanded it that other groups or projects who are using GBIF data can also have the local hosted portal triggered to their needs. I know in discussion with DiSSCo and others, there are plans to look at further needs and developments. But here we really have to discuss with GBIF which are their priorities and their roadmap, and then see how additional needs can be covered and so on. But for example, with our DiSSCo Flanders project, we also not just develop on our side, but we also offered to work together with GBIF and Tedwick and others on developing these standards. But it's something, this roadmap is not something that one of these groups can do in an isolated way, where we have to meet together with this big worldwide and European associations or bodies or initiative to a common goal, and then see what is feasible. Of course, it has a cost. So I cannot say that there is one roadmap. There are multiple roadmaps where we have to find common ground between these different associations and their priorities.
Thank you. Rod in the back, if you give him a mic. Oh, Colleen's right behind you.
This is a question for the last four speakers. So we've been talking a lot about citation, maybe citations at different levels, from whole volumes to individual articles, to bits of articles, to bits of data that are cited. And I guess there's two reasons for doing this. One is a scientific reason. You want to track provenance and give context to what you're looking at. And the other, of course, is metrics to impress people. Do we have any insight into which kind of thing we're measuring has the greatest impact with funders? What do they want to see? What numbers are most interesting?
I think this is not a no question. We don't know. We're at the beginning of that. So we have the DORA declaration for alternative metrics, which says we should look beyond just measuring the impact of journals and so on. But we are not yet there to have convincing models. Maybe Flanders has some more to say about this. What is really a manager or a director or wants to see? But they can also not decide because they don't really know what we can offer. So it's a little bit a chicken and the egg thing. I think we have to lead on that. We also know what's important for us. And we should actually make sure that these things are in this metric. And I think it should also be a measurement to not just who did what and all this human-centric thing. We should actually say what contributed to grow our knowledge graph, what contributed to better understanding our Earth. So we need definitely metrics which are for biodiversity and conservation and science.
Patricia, also, did you want to speak to this?
So in Flanders, they have S3 roadmap where we submit all these research infrastructures like DiSSCo, and they actually aligned with the KPIs that this S3 forum is putting forward, and then adding some of the local additional things they want to know, because basically it's about return on investment also. Not only, there is a part of fame, visibility, how many records come from Belgium and things like that. But they also want to see return on investment, who is using this information in other domains. The question of collaborating with the private sector comes back. And then there is the European Open Science Cloud, which is very much present now, who are also developed some kind of KPI to show how open we are, that they also follow. So they don't invent something local, but they use this global recommendation that comes from the EU or the overarching body, but then they tend to add additional local criteria they want additional to that. For DiSSCo Flanders, we had an interesting discussion with them where they even came back to my colleague to say, don't put KPIs that just look good, but that you know that you can't bring the figures after. So be very careful which KPI you suggest, and pay attention that you also can find these numbers and these answers afterwards, and not just look good on paper, but then at the end or midterm, you don't even have the numbers or the first idea what to say by that criteria. And those coming top down from the funding bodies or from the EU, many are unfortunately that they want numbers that we can't find easily. And so it's a dialogue to explain also why at some cases we can't give them the answer they await.
One thing I've noticed, and I've only seen it on database, but that I think might be useful that I would like to see is, I know that in Scopus, and I don't know how they've created the algorithm where they do this, but they tie articles to United Nations Sustainable Development Goals. So you can look at somebody's citation record and see which goals their research has hit, which seems like a way to potentially share how impactful it is to certain areas of research. Other questions or comments on this one? Okay, Colleen, do you have another?
I do have one more online. And this might be for anyone to answer in the room. From Claire Borsch. I am an artist using imagery from BHL for my installations. I would like to create visual artworks that the public could interact with through sensors that then reveals the data behind the visuals. For instance, where is the species endemic to? What is its status as a threatened species? What resources would you suggest I work with to add this interactive and educational element? Any suggestions from the audience?
Yes, I mean, GBIF was an obvious answer. I just want to write one story about GBIF. And the very first Ibn Nelson challenge that we ran, one of the entries that I really loved, that nobody else loved, was somebody submitted, basically they did 3D printing of the numbers of species in particular areas within a country. So you got essentially like a very sort of spiky map with very tall peaks if that country was very biodiverse. You could get a country with very kind of flat if we were either ignorant about it or it wasn't particularly biodiverse. So these were kind of things you could see and touch and play with based on occurrence records in GBIF. Now, it's probably not particularly visual, but there's lots of kind of really interesting things which you could do. Sadly, nobody else in the panel of judges liked it, and it didn't win much, but I thought it was kind of cool.
Other thoughts? Patricia again.
For a bit fancy visualization, it's not really in the natural science area. There is a EU initiative called timemachine.eu, where they digitize a lot of archives, books, but also artifacts in 3D. They also work with the gaming industry for serious gaming. And I was in one of those meetings. We also discussed the natural history time machine to see how it was before. And I can say they have quite cool visualizations, and we could approach them to have this kind of fancy virtual reality and other visualizations they do for artifacts to reuse also in natural sciences, where they can recreate a 3D environment, how it was in the past, and it's quite advanced. And they have the gaming industry somehow convinced with some good funding. So this could be a way if we want something more artistic and more for the general public, or more wow effect.
And I'll share from the online audience, BHL Wikimedian in residence, Tiago Lubiana, shared that he agrees with GBIF as a source, but he has also connected some things to images through a tool called BHL Image Explorer. I can share that. He has shared the link, trying to bridge the audience online and those of you here in the room.
Thank you. Any other questions? Okay, then at this time, I think we'll invite David Iggulden up to close us out.
Hello, everyone. Yes, in case you haven't encountered me before, my name is David Iggulden, and I am current chair of the BHL Executive Committee. And it falls to me really to wrap things up for today and also say some huge thank yous, particularly to start with to all our speakers today. Can we please give them a round of applause to start with? I'd also like to very much thank Museum for Naturkunde Berlin, and particularly Elisa Herman, who has been hosting us, or is continuing to host us all this week for the BHL annual meeting, and has done stellar work really in organising the symposium today. So thank you very much, Elisa.